Dr. Marianne, please.
Okay, good morning everybody. Muy buenos días a todos los que se conectan a través de YouTube. And good morning to our friends in England, very far away. It's good afternoon for them. It is my pleasure to open this event. Let me share my screen, and really, I would like to welcome everyone to this joint conference between Pontificia, Universidad, Católica, Madre, Maestra in the Dominican Republic and the University of Wolverhampton. Excuse me. Okay. Very briefly, I want to introduce our institution, which is a Catholic, private, nonprofit institution of community service. It was created by the Conference of the Dominican Episcopate in 1962. Pucmm, as we mentioned before, is the Pontificia Universidad Católica Madre Maestra. It's actually the leading university in research in the Dominican Republic by Webometrics ranking. It's the first university in the Dominican Republic to obtain an international patent. Actually, we have 137 academic programs from higher technical degrees to graduate, postgraduate, and also a couple of doctoral degrees. We do have two main campuses, one in Santo Domingo and the main one in Santiago. We are national accredited by the Higher Ministry of Education in the Dominican Republic. Today, we are going to share a very interesting program with our friends in Wolverhampton. We have, since this pandemic, we had the opportunity with the support of Eduardo León Jiménez Foundation. We have the opportunity to open this Centro de Investigación de Biología Molecular with the name of a very well-known cardiologist in the Dominican Republic, who was a very, an investigator, a very well-known investigator, Dr. Salomón Jorge. In there, we are able to carry out research in the areas of molecular and cellular biology, immunology, and microbiology. It's a biosafety level 3 facility, specially designated to create a safe environment, both within and outside the lab, for work conducted with infectious agents and toxins. We have flow cytometry, fluorescence microscopy, RT-PCR, multiplexing technology, among other equipments. I want to welcome our friends from the University of Wolverhampton, and at this moment I want to introduce Tracy Ward, so you can introduce your institution. And thank you all for sharing this moment with us at Pucmm. I think this is a historic day for us, really, believe me. Thank you.
Thank you very much, Marianne. And first of all, can I extend the thanks from everyone at the University of Wolverhampton, to our colleagues in PUCCM, and also Professor Armasilla for organising this online conference event today. We are very much looking forward to hearing about the exciting research that is undertaken at your institute in the Dominican Republic, and also sharing our latest research from the Research Institute of Healthcare Science. We are a large research institute within the University of Wolverhampton. We have over 70 academic staff that are involved in a variety of research areas, and a large postgraduate research student population of over 90 registered students at the moment. Our research is divided into four core research areas of molecular and clinical medicine. We are very fortunate to have a number of clinical professoriate that are members of our research institute, and that really helps us translate the scientific and qualitative research that is undertaken at the university into real benefit for patients. Our other core themes are communicable disease and infection control, pharmacology and pharmacy, and encompassing drug delivery and drug delivery methods, as well as drug development. And finally, public health, epidemiology, and delivery of healthcare and healthcare practice. I think there is a great deal of synergy with the type of research that we undertake at Wolverhampton and with your institute in the Dominican Republic. And I think there is opportunities for us to develop some really fruitful collaborations in these areas. So I'm very much look forward to hearing from all the presentations, both in from the Dominican Republic and also from colleagues here at University of Wolverhampton. And thank you very much.
Thank you very much, Marianne and Tracy. Thank you, Marianne. Sorry, when you were doing your presentation, I was trying to sort out the problem of Ian Nichols, and there might have been some signs. I don't know if they came across. I think I have sorted, but I am not sure with the problem with the video, but we will know anyway. So without further delay, we are going to start our very interesting program of presentations. The first part is going to be the program of talks dedicated to infectious diseases and immunology session. And then I'm going to chair this session, and the first speaker is going to be Professor Paul Kirkham from the University of Wolverhampton, that is going to deliver a talk titled Chronic Respiratory Disease from Pathophysiology to Biomarkers. We have 10 minutes, so if you can do it in 8, 9 will be great, because otherwise, because of the time difference, we don't have a lot of time left. So I encourage you to please keep it to 8, 9 minutes as possible. So thank you very much, Paul. Please start.
Okay, thank you. I'll just put my share screen on, and then I'll go to my presentation. Can you all see that?
No, not yet. Remember, if it is in your desktop, just push the green button that says share screen, and then
That's what I'm doing. Can you see it now?
Yeah, you have to click on the presentation, because you must be able to see your desktop.
Oh, okay, hang on.
Then you have to choose your presentation.
Oh, okay, that's what you mean. So I've got to click on that.
Right. And then it says share, and then you have to click again. I think it's a blue button.
Okay, click that.
You can click twice over the presentation.
Right.
Hold on.
Or click
Right,
twice.
can you see it?
Yes, but no presentation. Oh, there
Yes.
it is.
Yes.
Excellent.
Right, okay, hang on. I'll just put it onto presentation mode.
Excellent.
Right.
Right.
Okay, well, my name's Professor Kirkham, as Angel just mentioned, and I'm going to give you a very, very brief overview of the research that we're doing in our group. Usually I could talk for hours on this, but as Angel said, I've only got 8 minutes, so I better get a move on. So my group is basically involved in respiratory disease research, and I've been doing respiratory disease research for nearly 30 years now, both in industry and in academia. The key research interests in our group at present are looking at disease mechanisms in chronic respiratory disease. And we've currently got 2 ongoing projects. One is looking at inflammation and lung fibrosis in COPD and pulmonary fibrosis, in COPD and idiopathic pulmonary fibrosis. And the other one is looking at macrophage dysfunction in COPD, which leads to a lack of resolution of this disease, and in principle is driven by oxidative stress. In fact, both of these diseases are driven by oxidative stress, which I'll come to in a minute. A third aspect is looking at autoimmunity and lung destruction in COPD, and we currently have a potential candidate who may be taking on this project. A more recent project, which we've just published on, is the impact of a protein called fructosamine-3 kinase, which I'll come to explain about later, is protective. And we found that polymorphisms or decreased expression of this particular protein can lead to increased risk of cardiovascular disease in COPD patients. And lastly, something which I've been interested in for quite some time since my time in industry is looking at diagnostic biomarkers for COPD. And currently I've got a patent out, which is under clinical evaluation with an industrial partner. So what is COPD? Well, COPD is basically a disease driven by oxidative stress, and it can cause a variety of impacts, as you can see, leading to chronic bronchitis, lung destruction, small airway disease, airway remodeling. And the oxidative stress can be driven either environmentally from cigarette smoking, which is the principal driver, but more recently, airborne pollution from industrial gases and car exhaust fumes can also lead to this disease. At the same time, there are also cellular forms of oxidative stress. One of the principal ones is actually driven by dysfunctional mitochondria, which I'll come to in a minute. But the net result of oxidative stress is to cause something called carbonyl stress, which is basically the non-enzymic modification of biomolecules. That includes proteins, peptides, DNA, lipids, whatever. And when you modify these biomolecules, it can lead to a variety of pathological conditions, such as from impaired steroid function through to tissue remodeling, to pro-inflammatory signaling, even to mitochondrial damage, which is actually a feedback, which then creates more internal oxidative stress and can actually lead to a vicious cycle driving effectively the disease or the chronic inflammation. And as you can see, the outputs, as you can see, are the pathology at the bottom. The bits circled in red are just the areas which I'm particularly interested in at the moment. So one of the things about carbonyl stress is quickly, just to show you that in COPD, you can clearly see the levels are elevated compared to healthy controls, which are the non-smoking controls, and even smoking individuals that are asymptomatic and show no disease. Also, although they have slightly elevated levels, are not as high as those that you actually see in patients with the disease itself. So clearly carbonyl stress is correlated with disease severity. And in fact, what we found here is that the carbonyl stress in this group will actually cause autoimmunity. And this autoimmunity is actually corresponds or correlates with disease severity. So as disease severity gets greater from what we call gold 1 stage up to gold 3, which is the more severe form of COPD, we can clearly see that the amount of autoantibodies against tissue proteins goes up. And this was work which I actually published a few years back in the Blue Journal. And currently we have an outstanding PhD project or MSc project, which is actually looking at these antibodies potentially as biomarkers. Particularly what we would like to do is in nasal swabs, because it's very difficult trying to get bronchial lavage from patients in the clinic. And the nasal swab would be much easier. But we'd also like to see if there's linkage to the histocompatibility antigens. So that's one project which is at the moment about to start. The project which my PhD student is currently undergoing is to look at the impact of carbonyl stress on macrophage function. And in this case, there's a particular protein, which is a stress-related protein called HSP70, which, although is induced in an inflammatory response, can actually modulate the inflammatory response through a surface receptor called CD24. And what we believe, and what my student is testing, is that we think that when carbonyl stress actually modifies this protein, it actually prevents it from binding to CD24, and in so doing, prevents the resolution of an acute inflammatory response and turns it into a chronic response. And not only that, it changes the function of the macrophages from a pro-inflammatory, from an anti-inflammatory M2 phenotype to a more pro-inflammatory N1 phenotype. And this M1 phenotype is where they lack the function to phagocytose bacteria, apoptotic cells, and this can obviously exacerbate and drive forward the chronic inflammatory response that one sees in diseases such as COPD. So if we then go to another project, which one of my students has just graduated with, and we published, and we've recently just published this data, is going back to fructosamine-3 kinase. What we found is that this protein is actually protective and can actually reduce carbonyl stress levels. Now, this protein was traditionally found in diabetics, and we found that it, as Dr. Dunmore will probably talk to you later about, is that it seems to be linked to increased or decreased risk of cardiovascular disease, depending on whether you got higher or lower levels. But what we found in COPD is that in smokers, you get an induction of this protective enzyme, which is good. So it's actually removing the carbonyl stress, which has been driven by cigarette smoking. And so this is being protected. But in the COPD patients, we actually lose the expression. And as a consequence, this can actually help to drive forward the disease. And not only that, we've also looked at a cohort of patients in the United States, over 30,000, and we found that there are gene polymorphisms which are linked to a dysfunction in the fructosamine 3 kinase, which can actually increase the risk of cardiovascular disease in patients with COPD. So one of the projects that he did is that he was actually trying to validate this fructosamine 3 kinase as a risk factor for the cardiovascular disease. And obviously we'd like to take this further forward into a much larger population and start doing gene knock-in and knock-out studies to see how this can actually impact the risk of getting cardiovascular disease. The next one is the impact of oxidative stress on fibrosis. And this is a project which my postdoc at the moment, Dr. Eagles, is currently working on. And the principle behind here is that when you get dysfunctional impaired mitochondria as a result of oxidative stress, these dysfunctional mitochondria, generally speaking, are removed through the mitophagy pathway. So they're ubiquitinated, and then they're recruited to the lysosome, where they're basically degraded down, and they're removed. Now, the issue about impaired mitochondria is that they actually will, although they won't produce less ATP, they will produce a lot of oxidative stress instead. And this can be like almost like a feed-forward mechanism, basically exacerbating the disease. Now, one of the things we found is that in fibrosis, for example, we know that oxidative stress drives the disease, and we know it damages the mitochondria. But interestingly, it also increases the expression of DUB inhibitors, because the mitochondria are so important for the energy production of the cell. A cell doesn't want to remove its mitochondria. It wants to try and maintain the mitochondria, as many mitochondria as possible, to maintain its energy needs. So what it does is it induces the expression of something called DUBs, the deubiquitinases, which actually remove the ubiquitin from these tagged mitochondria for degradation. And so thereby, they're, in effect, what's the word I'm looking for? They're maintaining the level of impaired mitochondria, and in so doing, are maintaining the level of increased oxidative stress. So in other words, they're actually helping to drive the disease. And we found recently in our studies looking at immunohistochemistry that we could see the expression of these DUB proteins is actually increased with disease severity. However, what we've in collaboration with a company, we're now looking at inhibitors of these DUBs to see if we can actually increase the removal of these damaged mitochondria, and in so doing, hopefully try and allow new mitochondria to be produced, and thereby limit the amount of fibrosis that develops as a result of the impaired mitochondria. And I think that's about it. And to sum up, the last slide just basically sums up sort of where everything fits in. So I have always had a very big interest in oxidative stress and how it relates to carbonyl stress, that is a non-enzymatic modification of biomolecules. And we've shown that this can lead to mitochondrial dysfunction in COPD as well as in fibrosis. It can also drive comorbidities. It can drive autoimmunity in many different autoimmune conditions. It can also drive chronic inflammation. And in my case, because I've got an interest in the lung, it will obviously lead to lung destruction and pathology. And obviously, in collaboration with biopharmaceutical companies, we're actually looking at novel therapies to try and intervene at these various stages, as well as a patent for the mechanistic biomarker, which I discovered when I was at Imperial College. And I think that about sums it up. I think I've done it in 8 minutes, Angel.
Thank you very much, Paul. That's great presentation, very interesting presentation. Thanks a lot. I have opened in the chat asking for questions, because we won't have probably time after each presentation to ask questions directly. But if you have any questions, and please, you can write it in the chat, then the speaker will ask these questions directly, or in the end, we will have a slot for 10 minutes for questions. So now we are going to move to our next speaker, which is Sergio Fabián Mosquera, one of the organizers of the conference, together with Silvia. Well, actually, they have done practically all the work. I haven't done much. So Sergio is going to talk about evaluation of the pulmonary immune response in infectious disease. So please, Sergio.
Thanks, Angel. Can you see my presentation?
Yes, go ahead.
Okay. Let me to activate my pointer. Okay. Good afternoon in England, and good morning here in Dominican Republic. It's a real pleasure to be with you, sharing this moment and learning about investigation focus of the researchers from the University of Wolverhampton. Watching the excellent presentation of Dr. Paul, I believe that most of the presentation will show us progress about the projects that are being developed. In my defense, I have to say that I began my working relationship with my university, with Pucmm, the last year. So I haven't started any project yet, and I have no results to show you. However, my objective in this event is present to the line of research that I want to develop at the university. Okay, so I just wanted to say that, and I can start with my presentation. Okay, so during my master and doctoral studies, we tried to deepen our understanding about the function of the respiratory immune system. And for that purpose, we use the tuberculosis model. This interest is due the lung has a very special condition. For example, the respiratory immune cells are kept supreme constantly in the steady state, and they have different kinds of activation depending on the stimuli. It's not the same, the activation to face pollution as for an infection process, for example. So it seems to me a very interesting topic. As we all, the respiratory tract carries out the vital function of gas exchange. This function is done at the alveoli that is located in the ends of the terminal of bronchi. There, with the presence of the capillaries, can be carried out the remove of the carbon dioxide and the uptake of the oxygen to the blood. For this function, the lung has the second largest surface area of tissue in the human body, and therefore the lung has a very strong and constant exposure to the environment, including inhaled pathogens such as bacteria, virus, fungi, parasites, allergens, as well as resident microorganisms. So to control these pathogens and microorganisms and to keep the homeostasis, the lung has a very important set of immune cells, such as alveolar and interstitial macrophages, dendritic cells, innate lymphoid cells, 1, 2, and 3, and can be recruited T cells, B cells, neutrophils, monocytes, eosinophils, etc. However, when a pathogen enters to the lung, these cells cannot act with all their potential because can affect the gas flow. So for that reason, the alveolar epithelial cells constantly supprime the immune cells through interleukin-10, tumor growth factor beta, and CD200. Even the surfactant as surfactant A and D have immunomodulatory properties. So in this inhibitory environment, this inhibitory environment can change when a pathogen enters to the lung, and these inhibitory factors will decrease, and another stimulation factors to activate the immune cells will increase. So this topic is very interesting to me and draw my attention to the study. So for that reason, the line of research that I want to develop in the university is pulmonary immune response in infectious diseases. It's important to result that some pathogens take advantage of this special condition of the lung to establish their infection. Pathogens like Mycobacterium tuberculosis, Listeria monocytogenes, and SARS-CoV-2, for example. So try to understand this situation in the lung is very interesting to me. With this line of research, we pretend to know the ways as pathogens evade the respiratory immune system in order to generate strategies to counter them and know the potential of the pulmonary cells to keep the homeostasis and lung function. With this idea and studying understanding the most important health problem today, we got the support to develop this project. We hope to start this project the next month, and it's titled Profiles Evaluation of Immunological Biomarkers in Peripheral Blood and Excel Break Condensate Predictors of the Evolution of the COVID-19 Disease. The objective of this project is to establish immunological biomarkers with cells and cytokines to anticipate the oxygen saturation less than 94% and acute respiratory distress syndrome in newly hospitalized patients with COVID-19 disease. And another project that we are going to develop maybe in January the next year with my colleagues from my previous university, Universidad Antioquia from Colombia, is this titled Search and Validation of New Inhibitors of the membrane transporters of the MMPL family from Mycobacterium tuberculosis as new antimicrobial agents with therapeutic potential. The MMPL proteins are vital transporters for the mycobacterial physiology. And we have 3 natural compounds that have a strong affinity for these proteins. So our idea is to evaluate the ability of these natural compounds to inhibit the function of this protein. And if can do it, maybe these compounds can be used in multi-resistant tuberculosis cases. So we need a lot of studies for that, but that is the idea. So this is what I had to tell you. If anyone has any question, I'll be attentive, and thank you very much for your attention. Was very quick.
Thank you very much, Sergio. Thank you very much. Presentation very nice as well of all the global interest in your laboratory. I have one question, but I'm going to write it in the chat. So we proceed, and anyway, we will discuss later at the end in the 10 minutes. Okay. So we will proceed with the next speaker, which is Dr. Ali Clark. Alice is giving a presentation titled Solving 3D Protein Structures to Understand Infection and Disease. So when you want, Alice, please.
Can you see my presentation?
Yes, that's okay. Please
Great.
go ahead.
Okay. Thank you for the opportunity. This is really exciting, and I really enjoyed the first couple talks, and I'm looking forward to the rest. I'm going to talk about my area of research, and I've given it a nice sort of general title. I basically look at 3D structures of proteins to try and understand infection and disease. Let me see, I need to click here. So firstly, I'm going to give you a really brief whistle-stop tour of method I use. And it's kind of, I want to do this because I want to give you some appreciation of the structure that I'm going to show you, how I got to that stage, the sort of things that I would do in the lab to get to solve a structure. I would like to talk about both areas that I research in, but I think it's better to pick one thing and sort of focus in on that and give you a kind of a nice trip through that and explain that one structure properly. So I do research into pore-forming toxins from bacteria. I also look at proteins from the human eye lens. So you're looking at this presentation through your eye lenses, your nice clear eye lenses, which are working really well, hopefully. And it's really important that those eye lenses remain clear, otherwise you get cataracts. So I look at what goes wrong in the eye lens and the molecular basis for things like cataracts. That's my other project, but I won't be speaking about that. I'll be presenting this toxin structure from a bacteria. So this explains the method that I use. So we take purified protein, and this is in a tube here. So it's floating around in solution, all these different proteins, these proteins that are floating around in solution, they're mobile. But to image it on the microscope, which is the method I use, it's electron microscope, we need to freeze it. And we need to freeze it in a very thin sample. So we blot off the excess, and then it's plunge frozen into liquid ethane. You can see there it's gone into the ethane and it's put in a little holder. So we've gone from this kind of mobile solution state into this frozen thin ice, and that's what we need to image. So that's put in the microscope. So you take the little copper grid, you place it in a special holder that goes into the microscope. These microscopes are huge, 3 or 4 times the height of a person. So we're talking a really big thing. Then the holder goes into the microscope and we image it. That's the gun producing the electrons. These are electromagnetic lenses that are focusing the electrons on the sample. So then they hit and pass through the sample. So think more like X-ray rather than an image. The electrons pass through the sample and then are focused a second time by a second lens here. And then they hit a detector. So we've got these electron detectors now, which are becoming quite advanced, and we're able to detect directly the signal from the sample. So if we look at this little grid in more detail, focus in on one of those squares, for example, you see the holes in the carbon, and then within those holes is that ice layer that we talked about when we froze it. So here, if we focus in, you can see there's 3 different molecules. They're all kind of randomly orientated in whatever orientation they were when it froze, and we pass the beam over them and get kind of like a shadow, like an X-ray of the protein structure. That's a 2D image. Then what we need to do is get lots of different orientations. So this is the typical image you'd collect from the microscope. You can see some of them are sort of lying on their side, some of them are end on. This is a little hollow barrel. So if you can imagine, some of those views are looking kind of down the barrel and some of them are looking from the side. So we pick out these images individually. We do this computationally. And then you can see we've got all different orientations. But to process it into a 3D structure, we need to have them in the same orientation. So we rotate them so they're all in the same orientation. Then we average them together and you get a much higher resolution image because we average out the noise and we boost up the signal. We do this for all the different views. So some of those are end views, some of them are side views. And then computationally, we work out what view they came from. Are they a side view? Are they an end view? And we can get this 3D reconstruction. So this is all done kind of computationally. There's quite a lot of kind of computer work involved, even off the microscope. Then we take this data and model a modeling that explains the data. So we fit in the amino acids, and here you can see this is mainly helical protein. So that's kind of what I do day to day, and the structures that I'm going to be showing you, we went through this process. So I'm going to talk about a protein from the aerolysin family of beta pore-forming toxins. The monomer structure was actually solved in 1994. So quite a while ago, but it's the oligomer that does the sort of the business, if you like, was the active form of the protein. So that was what we really wanted to understand. So that's what we set out to solve the structure of. You can see other members of the family from different organisms are involved in different diseases or have different activities. Intratoxemia is what you get from epsilon toxin. That's a sort of a disease in sheep, so that has an agricultural relevance. But then we have things like this intratoxin from Clostridium perfringens is involved in food poisoning in humans. So they have different activities depending on the organism. Basically, the way they work is like a little hole punch. So they punch holes in the host cell's membranes, which kind of creates a leak, allows the contents of the cell to sort of spill out. It releases lots of ions and small molecules from the host cell and kills the host cell. And this sort of acts as a virulence factor for the infecting bacteria. So what we did is we took the epsilon toxin, we put it on cells, and you can see there, as we add more epsilon toxin, you get more cell death. And at about 100% cell death, we take that sample, run it on an STS page gel, and you can see that the pore form or the oligomer form has formed. It's stable to STS. It has very high stability. So we took this sample and we're able to solve a structure of it. So you can see here the different subunits are colored different colors, and you can see there's 7 of them coming together to form this pore. And you can see this bit kind of punches through the membrane, and we're looking down. This is the hole that all the contents of the cell leak out through. Then we modeled into that map, and this is the model that we got with the dimensions listed on it. We then were able to kind of look at it, compare it back to the monomer, and try and understand how the monomer might transition into the oligomer. And so what we see is that we think is that these monomers come to the membrane, start to assemble into an oligomer, and this activation peptide shown here in red is cleaved off, and this pre-insertion strand in the monomer then undergoes a really big conformational change and punches through the membrane, a lot like you'd see a hole punch. So this is our kind of molecular model of what we think is happening. This is the activation peptide, the one that was shown in red before, cut off. This is the monomer that was solved before we did the work. And then we are able to use our oligomer to sort of see how it might come together on the membrane, and then you get this huge conformational change, and it's literally those beta strands punch through that membrane and form that hole in the host cell's membrane. And I've just got it here a second time, just showing it with the monomer, so you can see how the monomer does it. So all of this is published if you want to go and see more details. It's been a huge amount of information in a really short amount of time, so you can go and have a look, and if you want to see more, it's published. And if you want to know more about cryo-EM, I really highly recommend this review by Nagales and Shears, or this lovely video by Gabe Lander, who I pinched a little bit of that for the video I showed earlier. And I welcome any questions. Thank you very much.
Thank you very much, Alice. Very, very nice presentation. Lovely to see all the structures. As you can see, Alice published this work in a very, very good journal. And we are collaborating now with Alice to try to establish the structure of one of the molecules we are interested in. So please talk with Alice to establish collaborations. Any questions, we will put it in the chat. I see that people are already using the chat for questions and suggestions, and then we will proceed to the next speaker, which is Silvia, another of the organizers, Silvia Callo, which is going to talk about characterization of resistant bacteria and a study of the synergy between nanoparticles and antibiotics for its elimination. Please, Silvia, when you want.
You are muted.
Yes, I'm sorry, I was muted. I was trying to unmute the microphone. Thank you so much, Angel, and thank you everyone for being here today. And I hope we get a lot of collaboration from this little conference. So my presentation is going to be a little bit different, just because we don't have funding for any project right now, and we even didn't have a lab until recently. So it's all of the data I'm going to present is in diapers still. So as my title said, I'm working on resistant bacteria. This all started like 3-4 years ago, when a couple of medical doctors that are specialized in infectious diseases came to our university to take a master's degree in biomedicine that I was teaching with some friends that are talking today too. So they explained to me that in Dominican Republic, there's no there's any surveillance program from the government to check the levels of resistance in the country. So as infectious doctors, they didn't have a clue what was the level, the basal level of the resistance. They didn't have a clue of the resistant mechanisms that were working in the country. So all of the information was coming from the US or from Europe, and they had to work with those guidelines. So if you see here, this is all the countries in the world, the surveillance programs they have, and Dominican Republic is this little island here in gray. So we don't have any information from that topic in the island. So they asked me if I could help them to start gathering information about that. So I agreed that it was really important to have a bit of information so they could work and they could decide which treatment it was best for each infection. So we didn't have money, as I said, we didn't have a lab, so we started small. Luckily, in Pucmm, we have undergrad students that are willing to do research because they have like a small period of research to get their medical degrees. So we collected some of those students, we could put them together and send them to 3 hospitals in Santiago, our city. So those were the only hospitals that had a record from the microbiology lab. Most of the medical centers of laboratories in Dominican Republic, they don't keep digital records. So they only have paper records. So we decided to check only the 3 hospitals that had a digital record the students could access. So 3 of the students went to the hospitals and collected pediatric services records, and like 8 students went to the hospitals to collect the adult records that were a little bit more. So we analyzed those from the hospitals that they use automatic machines to do the resistance screening and the identification of the pathogen. We have those 2 papers published in really small journals because they are really easy, they are simple. It's just analysis of the different levels of resistance from several different mechanisms. After that, we wanted to do actual research, so do a little bit of experiments. But as I said, the students only have like 3 months to do the experiments, and they don't get any money. So they are really small studies. The first one is almost in the editorial process. Is this one where the students collected isolates from urine that were identified as Escherichia coli. So they collected those isolates, went to the lab. We had a small lab, a nanobiology lab that Dr. Zaira Gonzalez is going to introduce in a little bit. So the students went there and just did a manual screening for SBL and MC enzymes. Those 2 give the bacteria resistance to the beta-lactam antibiotics. So they checked that manually, and that's it. We didn't have money to do more apart from that. They collected the reports from the hospital and did the manual detection. So it's going to be, again, in a really small journal. Then the two medical doctors, they also did a small experiment during the master's degree. These two are the medical doctors, and they only have also 3-5 months to do the experiments, and we didn't have a lot of money for that either. So we did the same. They collected samples from one of the hospitals. They did the manual detection for SBL and MC, but in this case, they also did PCRs to detect the actual genes that were expressing the isolates. We didn't have enough money to send them to sequencing, so it's only PCR to detect which kind of gene, the family of the gene. We cannot be sure of the specific allele that is expressing, but we can have at least an idea which are the families more commonly expressed in the isolates collected in Dominican Republic. So that one is also, we are working to finish it and send it to publishing at the end of the year. But it will also be in a small journal because it was like 54 isolates only. So that's all that we have until now. Then the pandemic hit, the students could not go to the hospitals to collect anything. So we are trying now to restart the researches. And we are also logically trying to get funding to do a longer study, because the problem with publishing these two small papers, the two at the end that are not published yet, the reviewers ask for longer experiments or a lot more of the isolates. But the students don't have more time or don't have more money to buy kits to screen. So we until now we cannot go further, but we are trying to get funding and do or try a longer surveillance program on our own. So check more mechanisms of resistance during more time, and also in different places, because we now focus in our city where the students live, but we want to collect samples from Santo Domingo or even the rest of the country, and maybe try 1 year gap of surveillance with each mechanism of resistance. And also because we have an nanobiology lab in the research center that Zaira is going to talk about in a little bit, we also wanted to try synergy between nanoparticles and antibiotics to try and eliminate those resistant bacteria we isolated from the hospital. So that's our ideas. We are trying to get funding, but at least now we have a really nice lab. So we have the Research Center for Molecular Biology, Dr. Salomon Jorge, as Marianne kindly introduced at the beginning. It's a really nice facility. We have a lot of equipment now. We have a real nice place with amazing views in campus. It's a really nice campus too. And we also have like a couple of culture chambers where we can manipulate the resistant pathogens without risking our students. So I hope with this lab, with nice lab, we will get lucky and get some funding in the near future. So thank you all for listening to me, and I hope we will enjoy ourselves for the rest of the event. I was going to say afternoon, but it's morning in Dominican Republic, so the rest of the day. So any questions you can write on the chat, and I will be answer them as soon as possible. Thank you so much.
Thank you very much. Very, very good presentation and very interesting, Silvia. I have seen very good equipment in the laboratories and EVOS microscope. I will indicate one of my pictures has been taken with the same kind of microscope, and I think it is very nice. I will tell you later in my presentation which one. The next one then is Wayne Heselvey. Wayne from the University of Wolverhampton will deliver the talk investigating novel treatments from acanthamoeba and leishmania infections. Please, Wayne.
Okay, can you see my slides?
Yes, thank you very much. Go ahead.
Okay then, hello everybody. So buenos dias to everybody in the Dominican Republic. Good afternoon to everybody in the UK. So as Angel just said, my name is Dr. Wayne Heaselgrave. And so just to tell you a little bit about me. So I've worked at the university probably for about 6 years now. I've got quite a large research lab with a postdoc, 6 PhD students. We do a variety of different things, developing ex vivo models by using pig's eyes to model the infection of acanthamoeba. We also look for endosymbionts to see how this relates to the disease itself, to see if patients that have acanthamoeba with endosymbionts have a poorer prognosis with the infection. We also sort of do work with cutaneous leishmaniasis. I'm not going to mention it too much in my talk, just because it's probably too much to get into 8 minutes. So I will just be focusing on acanthamoeba. We do lots of organic synthesis, in particular looking for novel biguanides, which can potentially be used as treatments. We also do look at novel ways in which we can deliver drugs. So we look at lipid nanoparticles and see if they can be used to deliver some of the drugs that we use to treat acanthamoeba. In addition to that, I do a large amount of analytical services for the contact lens industry. So working for companies like Alcon, Cooper Vision, doing all of their testing. So we test things against bacteria, fungi, and of recently testing things against coronavirus as well. I also work for the International Standards Organization. So we're a part of a team that are developing new standards for contact lens testing. Okay, so you've probably all never heard of acanthamoeba keratitis, so I'm going to tell you a little bit about it, but I will warn you that some of the pictures are not particularly pleasant. So acanthamoeba is an infection of the cornea. The parasite enters into the epithelium and then penetrates into the underlying stroma. Fortunately, it is very, very rare, but it is probably the most difficult eye infection to manage. There is no licensed treatment. You're probably looking at anywhere between 6 months to 2.5 years of treatment, which would be administered maybe 6 to 8 times a day. It's incredibly painful infection. In many cases, it leads to a reduction in vision and blindness, as you can see in the images on the side of the screen. Okay, so acanthamoeba isn't really a true parasite. It's a free-living amoeba. It's called acanthamoeba because it has these thorn-like projections all over its surface. It's a eukaryotic organism. So it's massive compared to a bacterium, so it's 30 to 40 times larger. And it exists in this trophozoite stage, this spiky stage you can see here, and it moves around in the environment due to these things called pseudopodia. What makes it interesting is it can transform into a dormant stage, and it's this thing that is the real difficult target when it comes to treatment. It's resistant to most drugs and most disinfectants at concentrations that can be tolerated by the eye. So it's a real big problem for patients. Okay, so there's a short video here that should just show you, this is an acanthamoeba moving around under my microscope. It's trying to get away from the light on the microscope, but you can see how it pushes out its pseudopodia in order to move around. And it can do this to move around on surfaces through the soil and things like that. Oops. Okay, so you're possibly asking yourselves, where in the world does this dangerous creature live? Unfortunately, it lives absolutely everywhere in the world. This isn't a tropical disease. It is a disease that you'll find in soil worldwide. I've collected samples from Costa Rica in the past, from South Africa in the past, and just from the UK. So you'll find it in all of your gardens. It'll be in your shower heads. It'll be in your sinks. It'll be in your vacuum cleaner dust in your house. So it's absolutely everywhere. It's almost ubiquitous in its distribution. So just to tell you a little bit about sort of what we do. So this is based on a paper that we published a couple of years ago with Moorfields Eye Hospital in London. And we looked at a variety of different things, but I'm just going to focus on looking at the efficacy of acanthamoeba treatments that can be given to patients in the UK, and how we're using this now to look for better treatments for our patients. Okay, so we started by looking at some of the treatments that are currently used. So at the moment, if you have acanthamoeba keratitis, you're prescribed one of 2 drugs. The first of these drugs is propamidine, which is given in the form of Brolene, and the other is a drug called PHMB, which I'll come to on the next slide. And propamidine in the form of Brolene has been prescribed for the last 20 years. Although we were able to show, sort of in this paper, without going into the results in too much detail, we were able to show that bromelain actually has no activity against Acanthamoeba whatsoever, even though it's been the mainstay of treatment for the past 20 years. Bromelain itself is available in an eye drop form. The drug itself, or the preparation itself, is preserved using a drug called benzalkonium chloride. And so what we did as part of our research, we used pure drug alongside the bromelain formulation. We mimicked the bromelain formulation, and we were able to demonstrate that one of the main drugs that's used to treat the infection actually has no activity whatsoever. All of the activity comes from the preservative that's contained within the preparation. The other drug that we're particularly interested in is the wonderfully named polyhexamethylene biguanide, which we usually abbreviate to PHMB. We looked at that alongside some other drugs like miltefosine, which is found use originally as an anti-cancer drug, but is now being used to treat a variety of different things, including cutaneous leishmaniasis. Drugs like macrolide antifungal drugs such as natamycin, and other antifungal drugs like prosaconazole. And we compared all these to PHMB, because at the moment there is no licensed treatment, and it is a very, very long protracted treatment regime should you be unlucky enough to come across this particular infection. Okay, so we were able to show that drugs such as miltefosine show good activity against both stages of the organism, including the dormant stage. Certainly sort of not quite as good as the PHMB. We showed that natamycin, the antifungal, only had activity against the trophozoite stage, but not the dormant cyst stage. But what was interesting, we showed the antifungal drug, posaconazole, performed to a similar level to PHMB against both the cysts and the trophozoites. And what's interesting about this drug is it has the potential to be given orally, whereas PHMB is used as topical eye drops. Okay, and this is just to show you what happens, just to show how this is on the left-hand side, you can see the healthy cyst. So what this is the trophozoite is encysted within a cellulose, very, very thick cellulose cyst wall. You can see the nucleus on the inside of the cell. After it's been exposed to PHMB, what will happen is it will perforate the cell membrane of the Acanthamoeba trophozoite inside the cyst, and you'll see leakage of the cytoplasm. You'll see a destruction of the nucleus, and you'll start to see vesicle formation, and it will start to shrink away from the cyst wall. So we know certainly that drugs like PHMB, which are disinfectants, seem to be the best option that we have for treating this particular infection. And so what we're currently doing at the moment, so I'm just going to focus on the organic synthesis side of things. So we're looking at drugs like PHMB, which was just simply discovered as a surgical scrub probably about 35 years ago. There was no rationale at the time. It was never intended to treat Acanthamoeba infection. In the UK, it's often used as a non-chlorine disinfectant for hot tubs and swimming pools. So what we're looking now at other drugs within this area, so drugs such as polyminopropylbiguanides, polyhexamethylene guanidine as well. So what we're doing is we're synthesizing the existing polymers, and then we're going to add different monomers into the sort of the growing polymer chain, and then to look to see what happens to these. So we'll characterize them using NMR and sort of mass spec. And then we're going to look at the antimicrobial activity against Acanthamoeba, but we will also do this against Leishmania as well. So if there's anybody in the UK or anybody in the Dominican Republic that has any unusual compounds that they think might have antimicrobial activity, please get in contact, because we're happy to test anything against this particular organism, because there is a complete lack of treatments for patients with this particular infection. So thank you for listening. So I just want to acknowledge Moorfields Eye Hospital, who we do all the research with, and the rest of my lab. So thank you for listening.
Thank you very much, Wayne. Very, very nice presentation. I'm sure there will be a lot of interest from our colleagues in the Dominican Republic. Please, when you are having a shower tonight or tomorrow, don't think about all the Acanthamoeba in the shower. Forget about that, but take all the rest. So now the next session is going to be chaired by Silvia Calo. So I hand over the control of the chair now to Silvia.
Thank you, Angel. So we are going to start the nanomaterials, biotherapeutics, and toxicity session. The first speaker is Taira Gonzalez. I named her in my presentation. She's from the Nanobiology Lab in Pucmm, but she's also working in Seville. So she will be talking about evaluation of the toxicity of nanomaterials in different organisms. If Dr. Wayne can stop sharing the screen, Dr. Zaira Gonzalez will be able to share her presentation. Thank you.
Hi, everyone. I think okay, I'm going to put here. Can you see the presentation?
Yes, everything is okay.
Okay, first of all, I want to thank both universities and the organizer for the opportunity of sharing our research today. I'm going to talk about what we are doing at the Nanobiology Laboratory. And this laboratory, okay, I'm having problem with the presentation. Okay, then this laboratory was inaugurated in June 2017, thanks to 2 projects funded by the Ministry of Health Education, Science and Technology of the Dominican Republic. And since then, we are working on the study of the interaction of nanomaterials with different biological entities. For those who are not familiar with nanomaterials, I want to remember that they are those materials smaller than 100 nanometers. They can have very different composition from metals or metal oxide or cholesterol, for example. And thanks to their nanosize, the properties of these materials are different than in the macro scale. They also have some advantages, such as their small size or the possibility of loading different drugs into them. So it is important to know the toxicity of nanomaterials before proposing new potential application. For that, in our lab, we use different model organisms of bacteria, yeast, eukaryotic cells, and also nematode. And we assess different parameters like cell viability, antibacterial or antifungal activity, survival, reproduction, oxidative stress, among others. We have some publication in that area in international journals of high impact factor, like Material Science and Engineering C, Toxicology in Vitro, or Chemosphere. I'm going to talk a little bit about this result, but not in detail. We demonstrated that the purification of the gold nanoparticle after their synthesis and also the growth determination method used can affect the antibacterial and antifungal activities of this gold nanoparticle, while the synthesis method shows no significant differences. We also demonstrated that the presence of zinc or nickel in ferrite nanoparticles may increase the toxicity of these nanoparticles and produce oxidative stress. We have assessed the in vitro and the in vivo toxicity of barium ferrite nanoparticles for the very first time. And these nanoparticles do not affect bacterial growth, could be ingested and accumulated in the C. elegans intestine. Our results indicate that this nanoparticle may produce toxicity in C. elegans, affecting both survival and reproduction, and especially in the younger larvae. We also work in our lab with other substances. Now we are evaluating the toxicity of the compounds of electronic cigarettes. For that, we are using the model organism C. elegans, and we are basically evaluating the survival, reproduction, oxidative stress. And in this case, we are also studying how the exposure to these compounds affect to a bacterial infection produced in these C. elegans. I'm going very quickly, sorry. I hope that you can understand me. We also have some international collaborations. We collaborated with Shandong University in China and University of Talca in Chile. We were evaluating the antibacterial and anti-cancer activities of different nanomaterials and also the toxicity of different green synthesized nanoparticles. And with these results, we have published some papers in also in international journal. You could see here some of them. We also worked with the University of Seville in Spain with myself for docetaxel delivery to prostate cancer cells. This work is finished, but not published yet. But this work has led to the application of an international patent. At the moment, we are working with the University of Seville in the designing of nanomaterials for drug delivery to the central nervous system, particularly targeting astrocytes and also microglia. It is very important because we have to avoid the BBB barrier, which is the most important barrier that protect our central nervous system. And we now are starting a new collaboration with the University of Córdoba, also in Spain, for designing nanoparticles for the delivery of microRNA to hepatocarcinoma. I'd like you to know that we are open to collaborate. Please do not doubt to contact us if you have any question, any suggestion, or if you are interested in our research. I want to thank again both universities for the opportunity of presenting today your research work, and thank you all for your attention.
Thank you so much, Tyra. Really nice presentation.
Thank you, Celia.
And next we have Dr. Laura Rubio, also from the Nanobiology Lab in Pucmm. And she's working with toxicological assessment of microplastics exposure on animal health, on human health. Sorry, I lost my image when the presentation came in. So Laura, the microphone is mute, so you can start whenever you want. Thank you for.
Okay. Can you see my slides?
Yes.
Okay, perfect. So thank you for the introduction. I am Laura, and I also work here in the Centro de Investigación de Biología Molecular, Dr. Salomón Jorge. And the area of expertise is in the field of environmental genetic toxicology. So I will try to explain you a little bit what we are doing in our group, but our main project is based or is focused on the toxicological assessment of microplastic exposure on human health. So in the next slides, let's see what happened. In the next slides, I will try to introduce you to the field. I will show you which is our main objective of the project, how we work, and also I will share you some conclusions of our first results. We have just started with our project. So microplastic contamination is the accumulation in the environment of the polymeric particles smaller than 5,000 microns. So this kind of contamination is rapidly increasing in all the ecosystems, as all we know. And the origin of these particles mainly come from the degradation of the accumulated plastic waste. So what does it mean? It means that we have two different kinds of microplastics. The primary microplastics are the ones that are synthesized in these small sizes with intended applications like biomedicine or even cosmetics. And then we have the secondary microplastics that are the ones that come from the degradation of the accumulated plastic waste. And this is a huge environmental problem. So anyway, the direct and the continuous contact that all we have with these particles, with humans, has arisen questions about the potential health effects, which we don't really know what could happen. So the generated microplastics could end up in different matrices, like food, for example, seafood or fish. Also could end up in the water we drink or the air we breathe. And we are continuously inhaling or ingesting this kind of particles. As I have said, we don't really know with which kind of environmental adverse effects we could suffer. So which kind of nature have these particles. So this the degradation of these polymers produce a very huge heterogeneous group of particles with different sizes, from macro sizes to nano scale, and different shapes. So we can find fibers, we can find pearls, flakes, amorphous particles, everything there. And also we can find different composition of the polymers. We can find in nature polyethylene, but also polypropylene or polyestyrene, and polycarbonate, polyvinyl chloride, etc. So our first shot, or our first try, we have decided to use the polystyrene because this is a highly synthesized and versatile material. It's used in different, so many applications, and it presents extreme resistance to be degraded. That means that it's going to accumulate, and in fact, it's accumulated a lot in the environment, and it has become one of the top 3 microplastics found in nature, which is a huge environmental problem. So what we know about the biological effects that this microplastic could perform? So for sure, we know that these particles are able to translocate intestinal and airway epithelial barriers. So we know that for sure. But the thing is that the available data that we have about toxicity, it's controversial. So in vitro and in vivo mammalian studies, we can find toxic effects, but also we have found studies showing non-toxic effects. So there is a lot of controversy here. So what we need is more studies on that. So which is our main objective? So what we want is to simulate a situation, an environment where we are inhaling or ingesting particles. So these particles will translocate the lungs and the intestine and will go to the blood and the lymph circulation through different organs. So we want to simulate this, but in vitro. We want to use in vitro cell lines like CACO2 for the intestine, BAS2B for the lungs, or THP1 for the monocytes in the blood. And then we want to check different parameters, some cellular or molecular parameters. I have just mentioned 4 here because this is our last study. We can measure cell viability, cellular uptake, intracellular ROS, genotoxicity, but we are open to collaborate and to test different endpoints. We have the capabilities and the facilities to test, like, for example, gene expression, cytokines. We can measure also cell cycle, apoptosis, etc. So how we work? We first need to do an acellular work where we characterize particles. We work because we need to be sure what we are exposing to the cell, so that which kind of shape we have and which kind of size. So which is our aerodynamic size. So for that we use the DLS and DLV to see the stability of the suspension. And then we are also using a transmission electron microscope to see how the particles look like, or which is the shape, and also the original size. And then we go through the cellular work where we could test different assays, as I have mentioned before. So I don't have time here to share with you our first study, but I can share you some conclusions, what we have obtained. We are just starting with this project, so we don't have not much to show yet. But what we did is we simulate this scenario, and we use 3 different cell lines, hematopoietic cell lines, and we treat with different concentrations of polystyrene, and we check these 4 endpoints. So, well, I remember you that we test cell viability, cellular uptake, how the particles are able to go inside the cells. We check also the reactive oxygen species generated by the particles intracellularly. And then we also check the genotoxicity by using the COMET assay. So what we saw is that the exposure of these 3 different hematopoietic cell lines was able to induce cellular effects. So ROS, and also we saw primary DNA damage. We also conclude that the effects are strongly modulated by the cell type. So this is the reason why some studies show positive effects and we can find also negative effects, because each cell, it will behave differently when they interact with microplastics. So it's very important to check different cell lines. Also, the underlying mechanism of cellular damage that we observe in the study is the objective of future study, because it's very important to know, to see the effects, but also to know which are the mechanisms by which these particles are doing its effects. And in general, what we could say is that this field has still a lot of gaps, and more studies are needed. We are just at the beginning, and we are excited to do more studies and to collaborate with someone who wants to work with us. So thank you very much. If you have any question, I will be happy to answer them.
Thank you so much, Laura, for such a nice and interesting presentation. Finally, for this session, we will have Dr. Bernice Wright. She will be talking about harnessing the power of the secretome to discover next generation biotherapeutics. Whenever you want to upload or to share your screen. Perfect, you can see it now. You have the microphone mute.
Presentation mode, or will it not let me? No, okay, that's fine.
You can go presentation
okay.
mode.
Great, okay.
Yes.
Great, thank you. All right, so my camera may go on and off because of connection problems, but hello everyone. Thank you very much, Angel, for this opportunity to talk about my research, my preliminary research, I should say. And it's been very nice to see what everyone else is doing here and in the Dominican Republic as well. So I work on cell secretomes, and this preliminary data shows my work showing cell bioprocessing of cell secretomes. And I can begin by talking to you about what the cell secretome is. So the cell secretome is proteins. It comprises of proteins secreted by cells as well as metabolites, and as well as extracellular vesicles that include exosomes, which are secreted from multivesicular bodies from that develop as intraluminal vesicles from multivesicular bodies, as well as microparticles and microvesicles from the outer cell membrane, and apoptotic bodies from dead cells or dying cells. And as you can see, I cannot, let's see, oh, sorry, go back. Okay, so you can see the size ranges of exosomes, microvesicles, and apoptotic bodies here. So exosomes are the smallest, and they range from 30 to 200 nanometers in diameter. And the markers, the positive markers for exosomes include tetraspanin proteins, so CD9, CD63, CD81 on their surface, and cytosolic proteins, ALEX and TSG101. And the markers for microvesicles include integrin and selectin, CD40, and we know the markers for apoptotic bodies are similar markers that you would look for in apoptosis, so annexin 5. And we have some information already, sorry, I'll go back, about the therapeutic functions of secreted metabolites and secreted extracellular vesicles that we can talk about later on in the slides coming up. Okay, so just to put the sizes of those extracellular vesicles into perspective, we know that exosomes are similar in size to viruses, and microvesicles are similar in size to bacteria, and apoptotic bodies are similar to platelets. And so this is a more detailed view of the production of microvesicles and microparticles from cells, and we can see here, if we focus on the lower image here, we can see that the mechanism by which intraluminal vesicles, so exosomes, are produced from multivesicular bodies include escort proteins, lipids, and tetraspanins. So we have some information about the biogenesis of exosomes, although we don't know exactly how these different components in the machinery within cells are involved in production of single and multiple multivesicular bodies that lead to the production of exosomes. Okay, so now just to talk about some of the cargo, the therapeutic cargo within exosomes, we are mostly interested in microRNAs here, and also some of the proteins that are found in exosomes. And we know we have some information about the therapeutic activity of the microRNAs within exosomes, which are the main type of extracellular vesicles that have received attention as therapeutics. And so now we need to think about why or how exosomes and cell secretomes and extracellular vesicles can be used as therapeutics. And so we know that that cell secretomes are being used as a source of biomarkers, for example, secreted proteins as biomarkers for certain types of cancer. So they can be used as diagnostics. And I found a recent paper, or maybe not a very recent paper, showing how proteins secreted from early tumors can be used as diagnostics for cancer. So we also know that there's a growing use of cell secretomes, particularly exosomes, as biotherapeutics. And there are some companies now that are involved in using extracellular vesicles as therapeutics for treating osteoarthritis, for treating rare pediatric disorders like dystrophic epidermolysis bullosa. And there's a global market for biotherapeutics. We know that this year it's been estimated that the market for biopharmaceuticals is at least $291 billion, and this includes biotherapeutics, because there are a number of companies now that are involved in using extracellular vesicles, particularly exosomes, as therapeutics, including Carousel, which is now using developing exosomes as cosmeceuticals, so as skincare, and AstraZeneca has been involved in looking at secreted proteins as therapeutics for a number of years now. Okay, so because of the industrial interest, the great industrial interest in cell secretomes as therapeutics, there is now a bottleneck for manufacturing high enough yields of exosomes or secreted proteins that can be used clinically. And because of that, this is the main rationale for this part of my research, looking at developing robust and efficient workflows for producing high yields of cell secretome products, particularly exosomes. So I'm focused on using skeletal myoblasts for producing extracellular vesicles and for producing secreted soluble proteins. So we're interested in using a cell culture system that involves the use of cell microcarriers, providing a physiological surface for growing those cells, but also for differentiating them, because we're interested in using the myogenic secretome to produce cell secretome products. And the reason for that is because we found some years ago that a cell line, a skeletal myoblast cell line, C2C12 cell line, was used to produce cell secretome that was then used for treating chondrocytes that were inflamed. So inflammation of chondrocytes is one of the major factors that lead to osteoarthritis. So this is some work that we're developing as well, developing cell secretome-based therapies for treating osteoarthritis. Although I will not be talking about that today. And so part of this workflow is purifying exosomes, or purifying, I should say, extracellular vesicles, and then identifying exosomes from this using a size exclusion chromatography technique. So we use the ISON column, and we can also purify proteins, secreted proteins, using this column. And the main analytics that we're interested in at the moment is to identify our exosomes, so identify positive markers and negative markers following the International Society for Extracellular Physical Research guidelines for identifying that part of the secretome. And so we also need to image, as per those guidelines, we also need to image our exosomes or our extracellular vesicles. And we're using mass spectrometry techniques, and we're using protein staining techniques to identify proteins from the exosome fraction and also from the soluble protein fractions. And I think those are the main points here, but what I haven't gone into here is the type of in vitro cell models that we're developing for us to test the therapeutic functions of our cell secretome products. Okay, so I'll just talk through some of our preliminary data. So initially we started by looking at a 5-day period for differentiation. So we differentiated our skeletal myoblasts in a serum-free differentiation medium. So serum-free because serum contains exosomes, and so we want to avoid any exosome that would contaminate our secretome. And we only want to consider the exosomes or extracellular vesicles that are secreted by our differentiating skeletal myoblasts. So what we can see here is that we don't have a lot of differentiation here because we can't clearly see multinucleated myotubes here, although we do see some evidence for differentiation. And the green shows a late stage marker of differentiation, desmin, and the blue is just staining for DAPI, the nuclei. And so for the 5-day period wasn't enough. So we went on to, and I haven't shown this here, to differentiate the cells for 7 days. What we need to keep a balance between differentiating the cells over a period of time. And so the cells can be differentiated, but the cells don't die because they're starved, because we don't change the medium. We want to look at what they produce over that period of time. So we then went on to look at, so we collected the secretome over that period of time, and we used our isone column to separate out our extracellular vesicles and soluble proteins. And we also looked at the whole secretome where we hadn't separated out those components of the secretome. And we looked at secretome where we removed all of the cellular debris, dead cells, and fragments of cells as well. So we have our uncleared secretome, whole secretome, our cleared secretome, our extracellular vesicle fraction, and soluble protein fraction. And we can see clear differences in the protein profiles here. So this is a silver stain gel that we've separated those proteins using SDS-PAGE. So this may indicate differences in possibly bioactive molecules, bioactive proteins secreted by our differentiated skeletal myoblasts. We then went on to think about how we could increase the yield of our myogenic secretome from these skeletal cells. And we looked at 2 different volumes. So we increased the volumes of our serum-free differentiation medium. And as you can see, with an increased volume of differentiation medium, we have an increase in proteins, soluble proteins that are secreted, and also in our proteins from extracellular vesicles, and proteins in our uncleared and our cleared secretome. And then we looked at increasing the number of days. Sorry, I can't really see the other side of the slide. We increased the number of days for differentiation. And we can see that with increasing the number of days for differentiating for the skeletal cells from 5 to 7 days, we have an increase in proteins, in the yield of proteins in our cleared secretome, uncleared, and extracellular vesicles and soluble proteins. And we also looked at the difference in the confluency of our skeletal myoblasts. So we increased the confluency from 50% to 80%, and we can see that we have an increase in the yield of proteins secreted with an increase in the confluency of the cells before we begin to differentiate them. So that was encouraging because it shows that we can, in our ongoing experiments, we can scale up the volume of cells that we culture, either on tissue culture flasks or on cell microcarriers, which we will progress to. And once we've done this, once we've achieved this scale up to increase the yield of skeletal myoblast secretome, we can then move on to our in vitro studies where we evaluate the function, the therapeutic function of our secreted extracellular vesicles and soluble protein, and also the whole secretum from the skeletal myoblast that we've differentiated. Okay, so I would like to thank my collaborators at the University of Wolverhampton, Professor Tracy Wall, who's also the second supervisor for my PhD student who generated this work, Amani Ragay, and also Amani works within the Science and Industry Research Center, which is a more commercial part of this work, and Professor Angel Amesela, who's also my collaborator here at University of Wolverhampton, and the bioprocessing scientist I work with at UCL. Okay, so thank you.
Thank you so much, Bernice, for being here today and let us know about those that interesting research you are doing. So this is the end of the nanoparticle session, and now Sergio is going to present the cancer session.
Okay, Sylvia, thanks very much. Thanks for Bernice for your excellent presentation. Very interesting topic. So the first speaker of the cancer session is Dr. Mark Morris. The conference is titled Investigating the Molecular Basis of Cancer Metastasis. And Dr. Morris, please. Are you mute?
I am muted. There you go. Can you hear me now?
Okay.
I couldn't share my screen and unmute at the same time. Let me start again. Apologies. Can you see my screen?
Okay, I can see it and I can hear you.
Fabulous. Okay, thank you so much for the introduction. So, my name is Mark Morris. I'm interested in a cancer geneticist. I'm also interested in epigenetics and general cell biology, cancer cell biology in general. Today I'm going to talk about some work we've been doing for several years now, trying to investigate the molecular basis, DNA changes and the associated protein pathway changes that are associated with cancer metastasizing to distant organs. In particular, we're interested in how breast cancer metastasizes to the distant organ of the brain. So we know that cancer formation and cancer evolution is driven by changes in the genome. Can you see my pointer here, this arrow? Can you see this? I can maybe turn this onto a laser pointer.
Yes, we
see the laser
see.
pointer,
Yeah,
okay.
no problem.
So tumor evolution is driven by mutations, changes in DNA bases or large chromosomal changes. And in addition to this, it's driven by epigenetic changes. The best understood of those is DNA methylation, switching genes on and off. So the basic idea is that normal cells pick up these errors, which gives a proliferative potential. And as these cells grow more, there's more likelihood of picking up more of these mutations. And the trick is, this is under a selective pressure of the local microenvironment that this tissue, this tumor cell is found in. So the environment is influencing which mutations give which advantage. So there's a selection on which mutations survive, all the way to the point where some of these cells which contain multiple mutations will then move away from the primary tumor into a distant organ. And then this new microenvironment of the distant organ provides a new selective pressure for these random mutations to be selected for. So you end up with a subpopulation of cells growing in a distant organ which contain different mutations to the originating tumor. And we assume that these different mutations are changing the biology of the cells and therefore enable these cells to proliferate in this distant organ. And if we can identify these, we can think about the biology and the treatment of these metastases. We've known for a very long time that certain tumors like to metastasize to specific organs, indicating it's not just random bad luck where these tumors find themselves. So lung cancer, breast cancer, and skin cancer most commonly find their way into people's brains. These are the ones that like to metastasize to the brain. And when you look at how quickly this happens, we also see that skin cancer and lung cancer generally metastasize very quickly into people's brains. So once you have a diagnosis of lung cancer or skin cancer, you may almost at the same time find you have metastases. Whereas breast cancer, the most common type of breast cancer, are these estrogen receptor positive breast cancers. They do readily metastasize to the brain. However, it takes a great deal more time very often for them to get to the brain, which is indicative that there must be more mutational points to enable them to evolve into a cell that is capable of proliferating to make these large tumors in somebody's brain. So we want to find out what these mutations are, because we think these will be the things that enable these cells to pick up a new phenotype to grow in this environment. So we think this is going to be driven by mutation and epigenetics. So we've looked at both epigenetics and mutation in tumors which have been taken out of people's brains, all of which originated as breast cancer. So it's all a metastatic breast cancer. So we have looked for abnormal methylation via methylation-specific microarray analysis, and we've also looked for mutations using whole exome sequencing in tumors which we've had taken out of people's brains, which are breast cancer metastases. So what we're looking for is something that changes, that occurs in the breast tumors that metastasize to the brain, but isn't found in the general larger population of people who have breast cancer. These breast tumors, the primary breast tumors that don't metastasize, we're looking for this difference in these two things, whether it be epigenetic or mutational. And the idea is that if we can find things which are specifically found commonly enriched in multiple tumors, in these metastatic brain tumors, whether it be epigenetic or mutations, this is an indicator. Firstly, it tells us of the biology, what's happening. But secondly, these may be a good prognostic marker, an indicator of who may get metastatic cancer. So lots of biology, possible therapeutics involved in this as well. So with our colleagues in the Walton Brain Hospital, they've extracted these tumors from people's brains. We've extracted the DNA and done this genome-wide analysis. We've done genome-wide analysis, methylation, and mutation analysis via exome sequencing. We then compare the data that we generated, the many millions of points of data that we got from this analysis, to publicly accessible data. So looking at the data in the Cancer Genome Atlas, which has looked at thousands of primary breast cancers for both mutations and DNA methylation changes, we can compare that to our samples. We've also looked in the catalogue of somatic mutations to compare our exome sequencing data with what is common and rare as a mutation in primary breast cancer. So we're comparing metastatic breast cancer to primary breast cancer in both instances. We're looking for significant differences. The epigenetic analysis we did a few years ago, and we've published this now, we've identified 10 genes which are epigenetically dysregulated, specifically in tumors that metastasize to brain. So they're enriched in these tumors. And it's an interesting list of genes. There are some transcription factors. I really like the biology of GALNT9. It's a protein that adds sugars to other proteins which are then secreted and sit on the surface of the cell. So it's changing the way these cancer cells interact with their microenvironment, their new environment of the brain. We've also identified a handful of microRNAs. And more difficult to understand exactly the biology, we've identified some long non-coding RNAs as well. These are a hot topic in the field of all sorts of fields at the moment. So we like the look of them, but we don't know quite what to do with them. So we've got some really nice epigenetic genes. Before I move on, really neat about epigenetics is it's such a sensitive technique, is we can identify these DNA methylation changes as a blood test. Basically, we can extract the blood from these patients, and the tumors tend to secrete naked DNA. We can isolate the naked DNA from people's blood samples, and then using a modified PCR method, we can see whether that tumor DNA is methylated in their blood. So we don't need actually the tumor at all. So we can compare these. So this is a nice idea that you might be able to develop a blood test in the future for these kind of markers. So moving on to the the exome sequencing data that we've generated as well. So we've identified 7 genes which are enriched, commonly mutated in these breast to brain metastases. Between 25 and 30% of all the ones we analysed have mutations in these genes. I haven't published this yet, so I apologize today that I've covered up my actual genomes. There are 7 genes behind this gray box, which are all really interesting. And one day I will reveal these to you, but at the moment I'm keeping them hidden. So the trick is they're very commonly mutated in these metastases, but in the general population of primary tumors, their mutation frequency is low. So there's an enrichment in these mutations. So we think these must be driving the biology that enables these cells to proliferate. So that's where we're at. We've got all these genes. We are just really trying to now work out the mechanism. So we're carrying out CRISPR knockout of certain candidate genes. We're using RNA interference as well. We're finding clones where we've knocked them out, and then we're doing microarray analysis to see which pathways are disrupted when we disrupt those individual genes. So we're kind of starting to tease out this data. We can see when we knock out some of these genes, it changes the physiology of these cells, the phenotype of these cells. This BNC1, when it's knocked out, the cells become more invasive. These little black dots are invading tumor cells through a membrane. So that kind of backs up that they might be important. So we're kind of at this point now where we found many, many genes, which we're now going to spend many, many years investigating to carry on thinking about more of the biology, building models of how these work within in vivo systems. So we're moving a little bit away from the genetics and into functional molecular biology and thinking about therapeutics as well. Years and years of work ahead of us now, many, many PhD projects just waiting to go by analyzing these genes. Okay, thank you very much. The epigenetics work was all carried out by Raj Pangani as part of his PhD. He's now graduated. The genetic analysis is part of Yvonne Oliveiras's ongoing PhD, and there were many other people who we've been collaborating with. I will stop right now. Thank you.
Okay, thank you, Dr. Morris. Very, very interesting, your conference. The next speaker is Dr. Tracy Ward, with the conference title Designing Novel Metabolic Therapies for Malignant Brain Tumors. Dr. Ward, please.
Thank you, and I hope you can see my screen. Excellent. Right, so thank you for the opportunity to talk about some of our research in neuro-oncology, and particularly about trying to design new therapies for malignant brain tumors. So I'm a neuro-oncologist, or a brain tumor geneticist by training, and I've been working in this field for longer than I think. If I say 30 years, it ages me somewhat, but so I'm just going to show you really a snapshot of some of our recent work where we've been looking at trying to harness the metabolic shift that a lot of malignant tumors have to look for new therapies. To give you just a little bit of background for those of you that are not familiar with the brain tumor field, in the UK, we have over 12,000 new cases per annum in brain tumors. They are the 9th most common of all cancers and account for about 3% of cancer cases. Now, this doesn't include the metastases, those tumors that Mark has just spoken about that start with a primary tumor elsewhere, and then the tumors travel and metastasize and seed in the brain. We don't actually even have any numbers for how many tumors overall do metastasize to the brain, but looking at about 6% of all types of cancers that metastasize, we know that this is a big problem. And certainly speaking to our neurosurgical colleagues, a greater proportion of their work is dealing with these primary cancers that have metastasized. And many of the same problems that we encounter in trying to teach intrinsic primary brain tumors are also applicable to cancer metastases. Now, there has been a 39% increase in incidence in the last 3 decades. This is probably due to an increase in effectiveness of diagnosis. So we move away from CT scans to more sophisticated MRI scanning, and also a willingness to diagnose, particularly in an older population. We do not know of any external causative agents for brain tumors. And this really then focuses our priority in trying to increase the efficacy of treatment rather than trying to prevent the tumours from arising in the first case. One important thing is that we usually think of cancer as a disease of old age, but in brain tumours, there is a significant proportion of childhood and younger patients that develop brain tumours. And in fact, brain tumours overall are the leading cause of death in individuals under 40 years old, and they're the highest average life years lost in patients. So one of our predominant areas is working on glioblastoma at the University of Wolverhampton, and this is the most common malignant intrinsic brain tumour with a very poor outcome, medium survival even with gold standard treatment of surgery, radiotherapy and chemotherapy with temozolomide is only 14 months, and the 5-year survival is very low at 3.1%. Why is it so difficult to treat these tumours? Well, firstly, where the tumour is located, there is a need to preserve normal brain function. And although there have been advances in awake craniotomies to preserve eloquent and motor function areas during surgery, this still becomes an issue. It becomes even more so when we think about how some of the biology of the tumour, particularly local invasion, the tumour cells break off and invade the local normal brain parenchyma and can be found centimetres away from the main body. So it's very difficult to estimate where the tumour margin is. The blood-brain barrier protects the normal brain from circulating compounds, but also prevents many drugs, certainly over a certain size, from reaching the tumour. And then we have the intrinsic heterogeneity with multiple clones in the tumour and inherent radio and chemo-resistant pathways, and also redundancy of signal pathways, which has really hampered the efficacy of single-agent clinical trials, many of which fail at phase 3. Just want to spend a minute talking about overall research strategy for developing novel therapies at University of Wolverhampton. We are lucky to have some excellent collaborations with neurosurgical and neuropathology colleagues. And we receive tissue through an ethically approved tissue bank with patient consent and anonymized patient material, although we are able to get appropriate clinical details such as outcome, length of survival and treatment. We receive our freshly resected tumour tissue from the operating theatre in fetal calf serum free media. This allows us to look, and this is another project which I won't have time to talk to today, that's been run by a colleague, Dr. Kesley Attridge, looking at what the tumour actually throws out and how it may influence and modulate its microenvironment, particularly with relation to immunosuppression. The freshly frozen tissue undergoes a variety of molecular profiling, both to identify potential targets, therapeutic targets, but also to make sure that they are present in the tissue and also in the in vitro models that we derive from this. And also to make sure that what we are using in our in vitro models, cell culture monolayers and 3D cultures, is actually representative of the patient in the tumour. We are also in the process of developing zebrafish models, and Dr. Caruso will be talking more about zebrafish in general a bit later on this afternoon. And of course, we can do various interventions, molecular manipulations, and drug screening, and look at the efficacy of what we are doing. So I just want to very briefly talk about one area where we have looked at targeting aberrant glycolysis or glucose metabolism in glioblastoma. And like most tumours, glioblastomas preferentially use anaerobic glycolysis to metabolize glucose, even in the presence of oxygen. And they're very, very glucose hungry. But what this does do is provide building blocks for growth promotion, and it also, an increase in lactate really enhances the environment to promote invasion, and also for the cells to survive in a hypoxic microenvironment. And the first stage of glycolysis is the metabolism of glucose to glucose 6-phosphate, which is driven by hexokinases. And this is normally done in normal tissue by the isoform hexokinase 1. But we found that HK2, hexokinase 2, was upregulated dramatically in almost all of our glioblastoma tissues, up to 1,000-fold. And actually, this overexpression is significantly associated with overall survival in patients. Patients with that have tumors with over 200-fold of HK2 expression had a much lower survival, median survival of 11 months, 3 months compared to 11 months in those tumors that had a lower level of overexpression. And we found that thank you, Mark, for giving such a beautiful introduction to epigenetics, that actually HK2, the expression of HK2 in the tumours is due to a reversal of hypermethylation. So in normal tissue, HK2 is turned off, and in the tumour, the hypermethylation is reversed, and we can see the average methylation is only 4.6% in tumours compared to 26% in the normal brain. And this is always good for a target, something that's overexpressed only in the tumour, not present in the normal tissue, is really the type of target that we want. And we see a very nice correlation between the levels of methylation and the levels of overexpression. When we do a CRISPR knockout of HK2, we can see not only does it reduce proliferation of patient-derived cell cultures by up to 44%, but more importantly, it potentiates or increases the sensitivity to temozolomide. And this is really the only effective chemotherapy drug that is used clinically at the moment. And we can see we have average effective doses of up to 60% lower when we treat in our HK2 knocked out cell cultures. So we've also tried to model the ketogenic diet. There is certainly anecdotal evidence that individuals who are on a high-fat, low-glucose diet respond better to treatment and also have a low incidence of brain tumours. And again, we showed that by replacing glucose with a ketone body 3-hydroxybutyrate, normal astrocyte brain cells were able to metabolize and grow the hydroxybutyrate in the red line at the same level as 3 millimolar glucose, whereas the patient-derived cultures, in no case were they able to actually metabolize this. And this gives another angle or another option for patients to think about how we might disrupt their glycolysis or their glucose metabolism in their tumors. We've also looked at drug inhibitions of various HK2 inhibitors, and again we find that the effectiveness of, in this case, bromopyruvic acid directly correlates with the levels of HK2 expression. And on this side of the graph, we have these very high expressing HK2 cell cultures, and our normal and our low expressing having a greater cell survival at various doses of the drugs. And we've also shown that bromopyruvic acid potentiates its cytotoxicity through apoptosis, and again the level of apoptosis induced correlates with the level of HK2. So we have a really nice target. We've done a lot of mechanistic and in vitro modeling with this, and just like Mark indicated with his work on the metastasis, we're now want to move into looking at this in vivo models and really get a better idea of the mechanisms and the effectiveness of inhibiting HK2 through various ways, and really thinking about how we can move this into a sort of clinical trials and trying it out in people. Oh, and there's my flow just come in. Sorry about that. So acknowledgements. It's a lot of this work has been done by various research fellows and PhD students over the last 6 years, with a lot of support from our clinical collaborators in Liverpool and in Preston, who have been very generous in providing tumor tissue and also neuropathological support, and also our funders listed down here. And thank you.
Excuse me. Okay, was in mute. Okay, Dr. Tracy, thanks very much for your presentation. Very, very nice presentation. Our next speaker is Angel Hermesilla. I have to say that Angel had the initiative of this approach, for which we thank you, Angel, for this opportunity. And Angel is going to speak about identification of novel therapies to treat cancer and cardiovascular disease. Angel?
Thank you very much, Sergio. I have prepared a very, very simple presentation as well. Well, our major interest for a number of years in our studies in the University of Wolverhampton has been the regulation, to study the regulation of blood vessel formation. We are particularly interested in angiogenesis. Angiogenesis, as you know, is a biological process by which new capillaries are formed from pre-existing vessels. And this occurs physiologically in the embryo, in the placenta growth, and the wound healing during the development. This is a very, very well-controlled process, and it is essential for the development of all organisms at the embryonic development and postnatal until adulthood. Obviously, with the development of new organs, new cells, these cells need oxygen and nutrients to grow and to form the organs, and that oxygen and nutrients is provided by the vessels. And we are studying the key molecules which are important in the formation of blood vessels at the physiological level in the development of zebrafish. This is a zebrafish. We have mentioned several times that we are setting up a very nice facility, zebrafish facility in our institute. At the moment as we speak, they are moving walls and planning everything, and we are going to have it very soon. And this is the vasculature of a zebrafish, which is a stain, and then you can see here, you can follow the development of the vasculature in the zebrafish during zebrafish embryonic development. But the problem with angiogenesis is that this very well regulated process become deregulated in some occasions. In some cases, this deregulation is going to be associated with pathology, excessive angiogenesis, or in other cases, insufficient angiogenesis. In the case of excessive angiogenesis, the tumoral cells are going to need oxygen and nutrients to grow, and then they are going to promote the formation of new blood vessels from pre-existing ones, from surrounding ones, which are going to vascularize the tumor and provide that oxygen and nutrients. If we can stop the formation of these new blood vessels, we can kill the tumor by starvation. And there has been a lot of research in this area, and new drugs have been developed and approved for the Food Drug Administration. The problem is that after a few months, around 6-9 months, the tumor evades the action of these drugs, which are directed only against one factor. We are therefore studying how to improve these methods, for instance, in glioblastoma patients, where this has been detected, to try to determine key molecules in this formation of tumoral angiogenesis that we can use to manipulate this process and to avoid the formation of new blood vessels. And we have one of the results that we are writing up a paper at the moment about one microRNA that we have seen exert a very, very strong inhibitory effect in angiogenesis. And then maybe we can use it. I have been talking with Zaira in the chat we can try to direct with nanoparticles this microRNA to the endothelial cells of the tumoral vessels, and therefore to avoid the formation of these tumors and kill the tumor by exacerbation. In other cases, we collaborate a lot with our colleague, which ran the clinical part of our projects, Professor James Cotton, in the Heart and Lung Center of the Wolverhampton Royal Hospital. And he is also professor of cardiology at the University of Wolverhampton. And then in this case, after myocardial infarction or critical limb ischemia, what we try to do is to try to promote the formation of new blood vessels. So we want to manipulate designing new drugs to block new blood vessel formation when there is excessive angiogenesis, and to promote new blood vessel formation when there is insufficient, like after a myocardial infarction. And then to do that, these are just a few examples that I have put here. What we do is to perform gain of function or loss of function experiments in human primary endothelial cells, in some cases by overexpressing one specific protein, and then to see the effect of overexpression of these proteins in the angiogenic process. We use adenoviral delivery. For instance, in this case, we have facilities as well to use recombinant incompetent viral vectors in our laboratories. And then in this case, you can see we overexpress specifically this protein in the endothelial cells. In other case, loss of function, we are going to do the opposite. We use siRNA transfection specific for one molecule to silence the expression of that molecule, as it is in this case. If you compare, we have completely blocked the expression of that molecule. And in other cases, what we do is to get endothelial cells from mice where the gene to express that molecule has been deleted. And then in these cases, what we do is to isolate endothelial cells from the lungs. The lungs are organs with a lot of vascularity, and therefore they are very rich in endothelial cells, and we have set up very well a method to purify the endothelial cells from these mice, and then we can see the properties of cells containing all the molecules, the wild type or deficient in one specific molecule, as in this case. And then what we do is to do experiments in vitro and in vivo. For instance, in this case, what we do is to see how the endothelial cells are going to form tubes in vitro. This is an experiment of matrigel assay. We seed the cells on a matrigel resin, and then after 24 hours, these cells are going to form tubular-like structures. That's what we call tubular morphogenesis. And then we see what is the effect of different substances in the formation of blood vessels. In this case, the formation of blood vessels is enhanced. We also look at the migration of the cells in the wound healing assay. We plate the cells and leave a gap between these two parts of the area of the surface where the cells are growing, and then the cells are going to migrate to try to close this gap. And then if we do this with cells that are wild type or that don't have one specific molecule, we can see the differences in the migration of the cells and in different processes. Also in vivo, in collaboration with our colleagues in the Centro Nacional de Investigaciones Cardiovasculares, CENIC, in Madrid, in Spain, we do these experiments to see the flow of blood. For instance, in this case, what we do is to block the flow of blood to the lower leg of a mouse, and then this is a normal leg, the flow by we measure by laser Doppler. Red is going to indicate the flow, and then you can see the flow, or this is the ischemic leg. We have blocked the flow by ligature of the femoral artery, and then we see how different molecules are going to restore the flow of those vessels. So this is to try to enhance the reperfusion of ischemic organs. not only we are interested in microvessels, in the small vasculature, but also we are interested in human aorta. We are interested in pulmonary arterial hypertension. I'm not going to go into details, but I am sure I will be very interested to talk with Sergio about the pulmonary studies. And then this is the picture that I was telling you before that we have taken with the EVOS. This is a healthy human aorta, and then we have labeled here with blue the DAPI, the nuclei of the aorta. Green are the elastin fibers of the aorta, and smooth muscle cells with the smooth muscle actin in red. You can see here all the continuous elastic fibers, but there is remodeling and degradation of all this structure of the aorta in a Marfan patient, a Marfan with aortic dilation, and then you can see here like the elastins are broken, but I think very, very nice picture, and then we are doing studies in this direction. We are trying to see using as well large scale differential gene expression analysis, the genes which are important in one specific process. In this case, it is during inflammation, and then we can see the genes that are expressed, differentially expressed when we knock down one protein, what genes are expressed when we knock down another one. And then with these informations, we do a bioinformatic studies to see what are the functions which are involved in the absence of this particular process. And this can give us clues to design new molecules where we can manipulate and we treat the patients that are suffering from this disease. So I would like to thank University of Wolverhampton, especially the Research Institute in Healthcare Science for the support, our funders, and especially the people. This is our colleague in the Heart and Lung Center, Professor James Cotton, that run the clinical part of our projects, and our group in the University of Wolverhampton who are doing all the experiments. Thank you very much.
Okay, Angel, thank you for your presentation. Very nice presentation. The next speaker is Dr. Ian Nicole.
Yes.
On the conference is the
Let's
road
see.
to the. Okay.
Yes. Let's see if I try. I'm going to try to share the video again.
Okay. Okay. Okay, but the conference is titled The Road to the Catenanes, from DNA repair to novel aspirin-derived drugs for cancer treatment.
Aston University, where they had the had a Mark 13 mouse model. And here you actually...
Can you hear it?
Yes, yes, but okay. You can...
Hi everybody, my name is Dr. Nico.
You can maximize the video.
My name is Dr. Nico. Yes. I wish to thank Angel for giving me the opportunity to present my research. I've had a particular interest in bowel cancer for some time, and bowel cancer or colorectal cancer is a very common cancer with high morbidity worldwide and in the United Kingdom. Now, of course, the vast majority of colon cancer is sporadic, but the subpopulation of individuals have a familial forms of bowel cancer, HNPCC, for example, or FAP. So I was interested in looking at DNA repair defects in individuals who carried mutations in the DNA mismatch repair genes. So we were interested in looking at in the Scottish population mutations in 3 particular genes, MLH1, MSH2, and MSH6. And these are critically important genes involved in DNA mismatch repair, and a subpopulation of people that have germline mutations, carriers of germline mutations in these particular genes, and at a much higher risk of developing bowel cancer. Now, when I was doing this research, it became evident that in the literature that there's increasing evidence that aspirin is protective against bowel cancer using epidemiological analysis. And this particular research has been expanded to show that aspirin is protective in individuals who are at high risk of developing colon cancer, such as HNPCC families or FAP families. So, of course, that raises the question as to how aspirin acts as an anti-cancer agent. Now, intriguingly, back in 2003, A.J. Goh and Richard Boland suggested that the reason that aspirin acts as an anti-cancer agent isn't just related to its activity as an anti-inflammatory agent, but actually it has the capacity to actually affect DNA mismatch repair through the induction of these repair proteins. So we decided to look at this in more detail. Subsequently, individuals suggested quite a different mechanism, that salicylates can actually affect the levels of a protein called P300. And so you can see there are a number of mechanisms that have been proposed for the anti-cancer activity of aspirin. So to understand the anti-cancer activity of aspirin, we actually collaborated with Dr. Chris Perry, an organic chemist at the University of Wolverhampton, synthesized a number of compounds that were related to aspirin to screen them to see if they had any toxicity specifically to colorectal cancer cells. And we identified a couple in particular, PN508 and PN517 did. In collaboration with colleagues at Edinburgh University, they actually added these compounds to STB480 cells in vitro. This is a colon cancer cell line, a DNA mismatch repair competent colon cancer cell line, and found that diaspirin, female diaspirin, PN508 and PN517 respectively, were able to avoid cyclone D1. So this is evidence that cyclone levels are being perturbed in the presence of these aspirin-related compounds. This was extended in vivo in collaboration with colleagues at Aston University, where they had a MAC13 mouse model. And here you actually put on the flank of the animal cancer cells that will grow over a 10-day period to have a quite substantive tumour on the side of the animal. In animals that were also injected with diaspirin, female diaspirin, you can see there's been an ablation or a significant decrease in the growth of tumours. Now, interestingly enough, the animals were able to eat well and drink well and were healthy even when exposed to these particular anti-cancer agents. So this is a relatively non-toxic mechanism for the prevention of cancer. We were also interested in looking at how these compounds interact with platins. So the platins, such as cisplatin, salplatin, and carboplatin, are used as anti-cancer agents. In particular, one of the mechanisms is they affect DNA repair by damaging DNA. And we showed that aspirin and female diaspirin, for example, in the STB480 cell line, is able to synergize, in particular with cisplatin and with salplatin. Salplatin is now a mainstay for treatment of colorectal cancer. So there's evidence that the aspirins and aspirin-related compounds can actually synergize with platins. This may have utility. We also were interested in the anti-cancer activity of aspirin with respect to the OE33 cell line. This is an esophageal cancer cell line. There's evidence that aspirin may be prophylactic with respect to the development of esophageal cancer. And here you can see that fumarate can synergize with cisplatin and to a lesser degree with salplatin as an anti-cancer agent. Now, in addition to this, we actually found that there may be evidence that salicylates can affect EGF. So EGF is epidermal growth factor. It's intrinsically incredibly important with respect to epidermal proliferation, epithelial proliferation. And EGF, when it binds to its receptor, is usually internalized inside the cell to perinuclear location. So we were interested in how aspirin might affect this internalization. So to do this, we actually had an assay where we actually took a fluorescently labeled EGF molecule, where we added it to the cells, internalized, and the EGF actually moves to perinuclear location. And this image here is a phase contrast image, and this, of course, is the fluorescently labeled EGF, as seen by confocal microscopy, after about half an hour of incubation with EGF. And you can see internalization of the protein to perinuclear location. Now, so that was our assay. So This is what we have here. I have these images showing you the effect of aspirin and EGF on internalization. And here you can see the presence of the EGF is shown in red, and the DAPI stains the nucleus. And you can see here that the EGF binds to the membrane of the cells. Upon warming, you have internalization of the EGF to apparent prolongation. In the presence of aspirin or female diaspirin or other related compounds such as diflunisal, the internalization is dramatically perturbed. Binding still takes place, and internalization can still occur, but it's slowed down. So this is evidence that aspirin may actually act to affect EGF internalization. Back in 2011, Greenspan also showed that ibuprofen and aspirin, for example, can affect phosphorylation of beta-catenin. They enhance phosphorylation of beta-catenin. So this is a Western blot analysis of extracts from SW480 cells incubated with these anti-inflammatory agents, and you can see there's an increased level of phosphorylation, in particular with ibuprofen and with aspirin. We were interested in looking at where this phosphorylated beta-catenin is found. Normally, one associates phosphorylated beta-catenin with the destruction. We actually find that phosphorylated beta-catenin is most likely to be associated with centrosome, and we seem to see an increase level of phosphorylated beta-catenin in the presence of aspirin and female diet. So what we're suggesting is that this is interesting. Why is this the case? So that beta-catenin is normally associated with the Wnt pathway, and we usually associate beta-catenin with signaling through the Wnt pathway. So this is an area of interest in the laboratory. Now, given that we become interested in beta-catenin, we collaborated with American colleagues to look at the structure of beta-catenin and association with alpha-catenin and with E-catenin. And to do this, these colleagues were interested in using a technique known as SANS, or neutron spin echo spectroscopy, and we did this at the IOL in France. And so SANS and NSC were used to look at scattering of the protein in solution. And so we initially looked at alpha-catenin, the purpose of this talk. So here we have the crystal structure of alpha-catenin as a dimer in the presence of elution. This is the crystal structure in green. In solution, the protein is remarkably elongated. And this work was further extended to come with a particular structure for the alpha-catenin dimer and its association with F-actin. And so we suggest that SACs and SANS can be used with NSC to look at the binding of the dimer in association with the alpha-catenin dimer in association with F-actin, and how alpha-catenin can act as a mechanism sensor. And so, to summarize, we're suggesting that aspirin has pleiotropic effects with respect to acting as an anti-cancer agent. We identified novel compounds that, based upon diaspirin, that exhibit a degree of specificity against human colorectal cancer cell lines in vitro or active in vivo, and these affected cyclin D1 and NF-kappa B, and that aspirin and salicylates can affect EGF internalization, and that aspirin and these aspirin-related compounds synergize with platelets may have utility in the clinic. And toward the end of our research, we became interested in phosphorylation of beta-catenin and also the structure of beta-catenin and alpha-catenin, and how these proteins can act as power transducers. And this is the list of the collaborators, and thank you very much for listening.
Okay, thanks to Nicole for sending us the video and to Angel for playing it. The next presentation is repurposing an old anti-alcoholism drug as an anti-cancer therapy by Dr. Canapans. Dr. Canapans, please.
Hi, thank you. Could you hear me okay? And I hope you could see my screen as well. Thanks.
Yes, very well.
Yeah, thank you. So I'm from Professor Wang's experimental cancer therapeutics group. So I'm representing the whole group here. So our group is interested in repurposing an anti-alcoholism drug that's been used in clinic for many years, and how we progress that as an anti-cancer therapy. So over the years, our understanding of cancer, we have developed quite a lot of drugs specifically, targeting cancers or, general chemotherapeutic molecules. But the problem always been cancer always progressing to a later stage, metastasis. So about 95% of the cancer patients, they die because of metastasis or drug resistance that eventually develops. So our group is interested in understanding what is exactly happening in this area and how to develop some effective therapeutics. And as a part of that, along with so many researchers around the world, we agree that tumors do contain a heterogeneous population, and some of the cells inside the tumor, driven by the tumor microenvironment, are behaving more stem cell-like, or also called as cancer stem cells. So these cancer stem cells are extremely resistant to any sort of treatment, and they can either survive the treatment and receive the tumor, or they have the ability to migrate from one place to another through specific pathways and spread to other organs to give metastasis. So with the risk of cancer increasing in the next decade, we still haven't addressed this issue. So this is where we are interested in. So we need new drugs that target this specific population, but drug development is not an easy task, because it would take about 15 to 17 years easily, and it's about $2.5 billion to develop a drug. So as a result of developing this drug, there is also a big risk of more than 95% of the drugs failing in the clinic due to toxicity. And as a result of that, we have a lot of pressure on healthcare system, because the cost of drugs keep increasing every year, and it increases by 10% every year. So there is a global drive for repurposing existing drugs, which will be a shorter route for development of drugs at a lesser cost, and we can develop more affordable drugs. So our group is interested in an anti-alcoholism drug called disulfiram. So disulfiram is used by several millions of patients over the last 60 to 70 years, I think. And it targets a wide range of cancers when it is combined with heavy metal ions like copper or zinc. That's because disulfiram, if you see the structure to the right-hand side, is made of two DDC molecules that has a thiol group in the core that can chelate heavy metal ions like copper, zinc, or iron. And when it combines with copper or any of the heavy metal ions, they generate extracellular reactive oxygen species, or they can either transport these heavy metal ions into the cellular cytoplasm and create intracellular rose, which eventually damages the proteins, the membranes, and important and interface with the pathways of the cells that cause cell death. So this is very specific to cancer cells, because cancer cells known to possess high amount of intracellular copper. And so we thought, if we could use disulfiram in that situation, so that will be taken up more by cancer cells compared to normal cells. And which is why disulfiram is also considered to be a very safe drug to be repurposed for various cancers. But the big problem we have with disulfiram is the currently available version of disulfiram is oral disulfiram. And when a patient takes oral disulfiram, it is immediately broken down in the liver into two DDC molecules, and the liver metabolism actually blocks the active thiol groups in the core. So these thiol groups, if it is blocked, they won't be able to bind with copper anymore. So which means there is no anti-cancer activity. So for this reason, Professor Wang's group and our research, we have developed various forms of encapsulated disulfiram, where we entrap the drug in the core of several carriers using nanotechnology, and we extend the half-life and stability of disulfiram in the circulation. So one example I would like to show here, although we work with different formulations, I'm going to focus on a PLGA encapsulated disulfiram. So we were able to extend the half-life of disulfiram to 7 hours in the circulation. And this will enable us to have disulfiram slowly released in the tumor microenvironment and it is very suitable for even local injections in the tumor for cancers such as mesothelioma or glioblastoma. So we developed patents around it, and we have a spin-out company, which is now progressing this as a treatment for patients. So I'm going to show you some data here because we work with different cancers. I'm just going to show one example data, say mesothelioma. So mesothelioma is an asbestos-induced cancer. Although the risk for mesothelioma in the UK has gone down and the US has gone down, globally, still asbestos use is one of the big problems. So the number of cases of mesothelioma still increasing and a lot of people dying. And over the past 40 years or 50 years, we haven't developed effective treatment for mesothelioma. Only a couple of drugs, pemetrexed and cisplatin, has been approved for treatment of mesothelioma. So we developed PLGA disulfiram as an orphan drug approach for mesothelioma. And we developed these microparticles, if you see from the images here. So the first thing we need to do is to make sure that there is no aggregation and uniform distribution of this drug, and also effective drug loading in our particles. And we also tested how much disulfiram can be released over extended period of time or a short period of time. So we verify how this drug works in a situation in an animal model or in an in vitro model, and we verify the stability. And then we tested these drugs on different mesothelioma cell lines. For example, the figure here, I've shown 4 mesothelioma cell lines, and we have seen very effective killing of mesothelioma cells of PLGA disulfiram in combination with copper. And it is able to achieve this effect in very low nanomolar concentrations. We are also interested in not only just progressing disulfiram as a monotherapy, we are also interested in a combination therapy, where if you see the red line is the current treatment, say for example, pemetrexed or cisplatin is the current treatment that is used for mesothelioma. So when we use in combination with disulfiram, it is effectively bringing down the dose that is required by the patient. So eventually this means there is less side effects. So if you see to the annexin apoptosis figure here, that shows when you use a low dose of disulfiram or the individual drugs, separately as monotherapy, we don't see much of cell death, but when we use in combination, it's much more effective, and that's another angle we are looking at. And a picture here that shows that this is a 3D spheroid model that represent the cancer stem cells in culture. If you see the currently used drugs like pemetrexed or cisplatin, after treatment for 72 hours, they will still reform the spheroids, which represents the tumor recurrence in patients. Whereas when you treat with the disulfiram, you can see the spheroids never come back. At just 1 micromolar concentration, we are able to achieve this very effectively. And another aspect of the stem cell, like I mentioned earlier, is the migration and invasion ability to move from one place to another. And this is mainly induced by tumor microenvironmental pressures such as hypoxia. So here we show that the mesothelioma cells, when we grow under a hypoxic condition, a low oxygen condition, they are able to migrate or invade much faster compared to the normoxic counterparts. But when we treat with a very low dose of 25 nanomolar, which is not even a cytotoxic dose, we were able to significantly block the migration invasion ability. And also, we understand from various cancer models that we use in the lab that hypoxia drives several EMT or epithelial to mesenchymal transition related markers, which are the key factors that helps the cells to migrate or invade. And after treatment with a low dose of disulfiram, we were able to reverse these hypoxia-induced effects on the stem cells. And finally, we use animal models. For example, in animal models, we want to see whether disulfiram is released from the drug into the tumor environment, and whether it is able to effectively combine with copper to form the copper DDC molecule. And we were able to achieve effective dose concentration in animal models. And here this figure shows there's a tolerability study. We have an empty carrier and also a disulfiram loaded carrier. And the animals tolerated this very well for about 20 days until all everyday injection. And also all the key vital organs like kidney, liver, lung, or brain is not affected by disulfiram treatment. So it's a very safe drug from animal model point of view. And also we showed a fluorescent model of mesothelioma here, where when we treat with disulfiram, a 5 milligram dose of disulfiram, we were able to effectively reduce the nodule formation of mesothelioma in the peritoneal cavity of the animal models. So this is just one example I've shown here, and we do this for various other cancers. And like I said, our group is not only working on disulfiram, we are working on various formulations of disulfiram or disulfiram and metal complex based drugs that we progress for different cancers like albumin copper DDC or zinc DDC or pegylated liposomes of these metal complexes as well. So I would like to stop here and thank our internal collaborators and our colleagues at University of Wolverhampton, Reese, and also our external collaborators and the funding bodies. Thank you.
Okay, thank you. Dr. Kanapan or Kanapans, without or with S? Is your last name?
It's without an S, yeah.
Oh, okay, okay.
But my email ID says that there is a yes.
Okay, yes. Okay, thanks for your presentation. Very nice presentation.
Thank you.
And the last presentation for cancer session is characterization of links between gut inflammation and the immune response in collateral cancer by Hafid Omar. Omar, please. I can't hear you. Are you?
Hafid, you are muted. Hafid?
Omar cannot.
Yeah, sorry about that.
Okay.
Yes, that's the classic mistake I always do. I never fail even once in not doing it.
No problem.
So thank you very much. This is.
Can you share the presentation, please?
I will share it in a second. Let me just put it. Hopefully now it will work. Share. Can you see my slides?
Okay, excellent.
Fantastic. Thank you very much. This is really good chance to show a little bit about the work that we are doing. It's a great opportunity. Thank you very much for organizing it and giving us this this chance. If on a way I can just tell you a little bit about my interests in research. These are the projects that are going in our lab, and I have 3 PhD students and 1 intern doing the work on 1 or 2 aspects of these areas. Apart from this talk that I'm giving, which is part of my interests, I'm actually also interested in microbiome and gut microbiome in particular. And I'm also interested in iron deficiency and its effect, again, on cancer. So in terms of inflammatory bowel disease, again, I'm interested in iron stores and iron deficiency in anemia in a pregnant woman, again, in relation to IBD, bile acid malabsorption and microbiome again. So basically, it is the mucosal immunology of the gastrointestinal tract. This is what I'm interested in. Anyway, here I'm talking about inflammation and the reason for that, because also I didn't list this in the first slide, but I'm also embarking on a new project that with a PhD student that we're going to look at the effect of exercise on cancer and mainly breast cancer. And one of the things that we were discussing is to look at the immune system or immunological aspects of this treatment, exercise treatment, to see if there are differences in the cytokine profiles. However, one thing going on around is the fact that whether inflammation, is it beneficial or detrimental in cancer? And this is been quite a long time debate now in the science community. So inflammation, just to go step back a little bit, as I showed here, they are these immune cells and the epithelial barrier cells as well. Now these immune cells, they produce cytokines, and this is something common with the epithelial barrier as well. And that is the epithelial barrier now is regarded as one of the, it plays a quite important role in the immune system in the gut. So all these cells, they share one thing in common, which is cytokine production. And these cytokines depend on the cocktail of these cytokines, or the composition of these cytokines individually, that will shape up or will dictate the tissue milieu, whether if it's disease or in health. So again, it actually shapes the tumor microenvironment as well. And in particular here, all these projects that I've told you about earlier on, I am interested in one kind of cells. For my sense, it's dendritic cells. So the dendritic cells are the educators of the immune system, and they prime T cells and B cells. More so than macrophages, because they are more efficient than macrophages in priming the immune system or teaching the immune system. So all these cytokines depend on the composition. As I said, either they would cause tissue damage, maybe tumor growth and metastasis. Here is the tale of the two stories, and this is what. So this is not even a conflict in the literature saying that so the tumor. One side produces anti-inflammatory cytokines, and these anti-inflammatory cytokines or produce other molecules which they actually damp down the immune system, so it's immunosuppressant, and that to protect itself from immune surveillance and protect itself from immune attack. The other side is the tumor produces pro-inflammatory cytokines, and it's inflammatory entity that does that to grow, to use it as, because most of these pro-inflammatory cytokines, we call them pro-inflammatory, but they are actually, most of them are growth factors, and the tumor utilizing that to develop. So here I'm going to tell you a little bit about work that I've done, because I'm using this at the moment to shape up the project that I will be embarking on. But at the same time, I am very happy to collaborate with anybody who's interested in any of the projects that I showed earlier on, or in the exercise treatment project. My interest, what I'm working on, is dendritic cells. I love these cells. I've been working on them for quite a long time. As I said, they are the educators of the immune system. So what happens, they teach T cells, they tell T cells what to do and where to go as well. So they give them a postcode, so they will home back to the place where dendritic cells encountered antigens or pathogenic antigens, and they don't go somewhere else. So they give them a precise site of the inflammation or the cancer in the case. So I don't know about you people, but if you are in Star Wars, I describe them as the Yodas of the immune system, because they are the wise guys of the immune system. So on that, I just wanted another important side of this, is that the immune system is tightly regulated and is tightly balanced. So if there is what we call immune tolerance, I'm going to explain this a little bit later, because this maybe it is a little bit of misnomer. On the expense of immunity, and again, it's immunogenicity, not immunity, because immune tolerance is immunity, that will lead to diseases. One like example here is ulcerative colitis. Ulcerative colitis is a pre-malignancy disease. So people not treated with ulcerative colitis, they go to develop colorectal cancer. On the other hand, if the immune tolerance increases, then that will cause autoimmune disease like a Crohn's disease. So ulcerative colitis and Crohn's disease are part of inflammatory bowel disease. Crohn's disease, there's a few evidence here and there saying it could cause cancer, but it's cancer with a small c, because it is a horrible disease on its own right. It's too inflammatory that it gnaws through the tissue, and it doesn't leave a chance for a cancer to grow. This is what we think. However, ulcerative colitis is the one I'm going to concentrate on, because I'm going to compare it to colorectal cancer as a pre-cancer disease. So I looked, as I said, at dendritic cells and looked at markers which they are, if dendritic cells expressed ILT3 here. Hopefully you can see my cursor, the arrow. So ILT3 and DC sign are markers for immature dendritic cells. So if dendritic cells express these markers, it means they are inactive. They do not induce an immune response on T cells. And it seems like to be an inactive ulcerative colitis, because ulcerative colitis, if it's inflamed, you can see quite higher levels of both anti-inflammatory and pro-inflammatory cytokines together, or molecules as well, activation molecules. So I looked at inactive UC because it may show you a better picture there. And in that, we found out dendritic cells are inactive. Now, I looked at CCR7. CCR7 is quite important molecule here because it's a receptor. If it's expressed on dendritic cells, it means dendritic cells can migrate to lymph nodes. It's a migratory marker, and it means that they can induce immune response on T cells. Where do they do that? They do that in the lymph node. So the lymph node is almost like a classroom where T cells get educated by dendritic cells. Now, I found out in this one that inactive, again, CCR7 is quite low levels. So it means dendritic cells are non-migratory and they are not active in ulcerative colitis. Now, I looked at toll-like receptors, and mainly toll-like receptor 2, and toll-like receptors are antigen, they are battle recognition receptors, so they recognize bacterial products or microbe products. So that would be lipopolysaccharide or ligands from the bacteria. Or so toll-like receptors, for example, when they bind to their ligands, they induce a cascade of events intracellularly, and that will lead to the induction of NF-kappa B, and that will increase inflammation. So here again, we found that toll-like receptor 2, which is recognizes quite many the range of bacteria. They call them it's a gram-positive bacteria, and that's still, even compared to inflamed, as you can see in the middle here, it's the levels are very low, and they are similar to that in healthy controls. These markers now is the other way around. So CD86 and CD40 are activation markers. So if dendritic cells express them, it means they can activate T cells in turn. And here again, in ulcerative colitis are very low. But if we look at the CRC in colorectal cancer, it's quite, they are higher than even in control. So dendritic cells here in colorectal cancer are active. In pre-cancer, they are sitting in their place, they don't migrate, and they are not active. Now, again, this is just to look at whether CCR7 expression changes according to the tumor size. And this is the tumor, and again, even to the activity of the tumor itself, if it's metastasized or not, because as Mark mentioned earlier on, in terms of metastasis, CCR7 is very, very important molecule. And in terms of dendritic cells migrate, but also tumor cells and T cells as well. So tumor cells express CCR7. And it seems like every time when the tumor is bigger, it will loses the expression of CCR7. Maybe that's important in terms of dendritic cells, and either tumor cells are not migratory when the cell is at stage 4, because it doesn't need to metastasize, it's already metastasized anyway. Or maybe because it's kind of involved the lymph nodes, and therefore there are no more cells coming back again. So that could be the reason. Now, this is quite an interesting experiment that we usually do, which is called mixed leukocyte reaction. And this is we mix T cells, so there's naive T cells, we separate that from blood, and we add different concentrations of dendritic cell from gut, or either UC or CRC from the tumor. And what we found out that this is—
Sorry, Hafid, if you can summarize, please.
Yeah, but everybody went over time. So anyway, thank you. So, and then again, they are similar. Systemically, inflammation is quite high, as we can see with TNF-alpha and interferon gamma, who are pro-inflammatory cytokines. Now, to conclude on all of that is that immunity and immune tolerance are this balance that exists between the two. It can't be immune tolerance. It seems to be like more of unresponsiveness rather than immune tolerance. Immune tolerance is good. We need more of it. So here is, for example, future treatment targeting that. In early pre-cancer, we can use immune induction in terms of treatment. In the later stage, maybe you need immune suppression. I know that other people went over time, but thank you very much. These are the people that I'm collaborating with and our funder here. And sorry, you can read the papers, they are published, and you can ask me any questions with emails if you want, and I can explain in details.
Thanks very much, Omar. Very nice presentation. So we have finished the cancer session, and we will start the last session of this intense academic day, and Angel will be in charge.
Okay, thank you very much. So we have two final speakers. Sorry, Hafid, because yes, we are running out of time, and we didn't want to go over 5. So now I am being a little bit more strict than at the beginning.
Yeah,
sorry,
but I was still within time, Angel, and it wasn't me the one who done that. You could have.
No.
But thank you very much anyway.
Not at all.
But I'm happy to collaborate. That's the main thing I wanted to say.
So our next session is for kidney and neurology disease. And then Evie is our next speaker. The presentation will be shedding light on the molecular mechanisms of polycystic kidney disease. Please, Evie, when you want.
Good afternoon, everyone, or good morning, depending on where in the world you're at. Can you see my slides? Because I'm not really sure.
Yes, but not in full presentation.
Okay, so let me put it in presentation mode. No one's gonna do it. Yeah.
That's all right. Thank you very much.
Great. So I'm going to switch gears quite significantly and talk about rare disease. But the good news, as this is the end of the session, we're heading towards the end of the session, is that I've kept it very short and sweet. So I'm just going to give you a snapshot of what we do in my lab. And if you want to discuss this further, feel free to get in touch. This is my email address. We are particularly interested in rare disease because, for a number of reasons, actually. One of which is that they're quite understudied and substantially misdiagnosed. So there is an association between almost postcode and location and what diagnosis and treatment you get as a patient for your rare disease. Our particular interest are ciliopathies, and very briefly, cilia. If you're not familiar with cilia, I want you to imagine them like the sperm flagella, so the tail part of the sperm. The difference there is that cilia are not necessarily motile. Some types of them, called the primary cilia, are immotile, and we find them in the kidney. What they do is that they help cascade a number of signaling pathways, one of which is Wnt signaling that we study in the lab. And don't worry about the different names. What I want to show you there is that many components of signaling pathways localize to the cilia or require the cilia for correct function. And what happens when this localization cannot be achieved, or when cilia don't form right, is that a number of diseases arise that are collectively referred to as ciliopathies. And in the context of this signaling pathway that has a canonical and a non-canonical part, what the consensus rather is that the primary cilium, when it forms, enhances the non-canonical pathway and shuts down the other cascade. So I want us to focus on one particular rare disease called autosomal recessive polycystic kidney disease, which is a rare ciliopathy. It has an incidence of 1 in 20,000. It doesn't appear to have any recognized gender bias or ethnicity bias. So we find it in both genders and all ethnicities with equal incidence. But what is interesting for us, being quite focused on genetics, is that it has very diverse manifestation. There's 3 different types. There's the severe form that is found in utero and usually results in death. And then we have the more conventional manifestation that is usually diagnosed around birth or straight after. And then we have the milder manifestation that doesn't manifest until the teenage years or later on in adulthood. So in terms of the symptomatology, what you can see there is that the individuals carry they actually carry cystic kidneys on both kidneys that progressively increase in size, kidneys lose their function, and the patients will eventually need kidney dialysis or hemodialysis or transplantation. And in the case of ARPKD, I should say that besides a kidney transplant, for some individuals, a liver transplant is also required, meaning that there's quite a lot of pressure on the individuals, their families, and healthcare systems. From a genetics perspective, what you see there is that there is a median age of diagnosis at birth. And you can see the range of the symptoms, but I hope you can appreciate that I have percentages next to them, which means that not all individuals will manifest these symptoms. The majority of them will manifest hypertension, and many of them will require a renal transplant. But when this will happen is not exactly clear. And obviously that's quite important information for the patients, because if they cannot have an insight on their diagnosis, it is particularly affecting their day-to-day life. So if we look at the genetics of ARPKD, we conventionally, up until 10 years ago, everyone was telling me there's only one gene that causes ARPKD, and that's PKHD1. We now know that there is a second gene, and this is called DZIP1L. What is characteristic about both of them is that they both have a relationship to cilia that I mentioned before, in that either they localize to cilia or they are an important requirement for cilia function. Most cases of ARPKD are caused by mutations in PKHD1, and DZIP1L has been associated with the moderate manifestation of ARPKD. Now, the conventional wisdom is that different types of mutations are associated with differing outcomes, in that 2 truncations would give rise to the more severe phenotype that I mentioned before, that usually results in death in utero, whereas milder mutations, milder in brackets, of course, that is 2 missense mutations would have a milder manifestation, and they would lead to individuals not needing intervention until probably their teenage years. Bear in mind that conventionally most individuals need some intervention by the age of 5. So this information that I've shown you there, I've told you it's conventional. However, it has been disputed in a number of cases. And what we are interested in as a lab is to try and dissect the molecular mechanisms of the disease so that we can have further information on diagnosis and also impact prognosis. Because the sooner we can have an idea on prognosis, the better we can manage the disease. I should also say that there's no pharmacological treatment for the disease. So these children, they're usually children, main option is to monitor their kidney function until the point where they will need either kidney dialysis, hemodialysis, or transplantation. So we are trying to address this issue of associations between molecular mechanisms and disease severity by looking at cells, looking at mouse models, and also we have access to a unique cohort of PKD patients. our first, so I'm gonna show you snapshots of some of our approaches. Our first one is to use this cohort of DNA from human patients, they're patients, so they're human, to actually try and sequence using whole exome sequencing and identify, actually validate or disprove this relationship I told you before between the different types of mutations and the severity of the disease. What I'm showing you on the right-hand side is the algorithm. You don't have to worry about it too much. And I'm also showing you how we identified some variants of interest. This is work in progress, and I think our current message is that the landscape remains pretty confusing. So for the patients that we have access to, this clear relationship between truncating mutations and disease and severe disease hasn't been identified. And this is something other labs have seen. So watch this space. I might be telling you something radically different in a year's time. What we also want to do is use the information in terms of the gene expression of each of these patients to see whether we can impact on their prognosis and also inform their treatment. And again, thanks to this unique patient cohort, we have identified substantial differences in gene expression between the mild, moderate, and severe patients. I'm not going to talk about names today, but looking at those looking at this figure, you can appreciate that there are some differences in gene expression compared to normal. These are kidneys taken from patients and normal individuals, and we are characterizing this relationship further. And finally, on the animal models, we have access to a number of animal models, and we've also created our own model because, again, an area that is kind of confusing in PKD is that so far we don't seem to have one amazing animal model that fully recapitulates the disease. So what we do as a research community is to try and use information from a number of models and interpret it in a meaningful manner. This particular animal that I'm showing you was generated thanks to CRISPR technology. And we have studied it at different stages of development. So early on to see whether it mimics severe, and later on to see how well it recapitulates moderate and mild disease. And what I can tell you so far is that the kidney phenotype is quite interesting. It does show signs of moderate PKD, and we're studying this further to see if, when challenged, it would aggravate the phenotype. So that's all from me. What I'd like to finish off with is thank a number of individuals, and of course Rhys and the University of Wolverhampton. But I hope you can appreciate that it's quite tricky to work with a rare disease, especially in the COVID-19 pandemic. And I'm happy to take any questions in the end or link with you further. Thank you.
Thank you very much. A very interesting presentation. And because we are running a little bit late, we are going to go with Jordi's presentation, and then probably we will have to do the questions by email later because we don't want to go too much over 5. So Jordi, please, when you want.
Yeah, thank you. Hi, can you all see my presentation now? Yeah, yes. Good, great. Yeah, good afternoon, everybody. Buenos dias. My name is Jordi Cayuso. I recently joined the University of Wolverhampton, and I'm coming from the Francis Crick Institute in London, where I finished my postdoctoral studies. So today I'm going to talk to you, I will not show you lots of data, I will be quite brief, but I want to discuss and show you the ideas that I want to put forward for the research projects I will be taking on at the University of Wolverhampton. So I'm particularly interested in understanding the cellular and the molecular mechanisms of tissue regeneration and neurodegeneration. And in particular, we want to understand how cell cell-to-cell interactions that occur during tissue inflammation influence tissue repair and neurodegeneration. So inflammation, we know it's very important in tissue repair and motor neuron diseases. In regeneration, the interaction between the cells in the immune system and the tissues that are being repaired is important because it determines the outcome of the repair process. So this outcome can be a scarring of the tissue and losing the functionality of this tissue, or full regeneration. And on the other hand, inflammation is also very important in the pathogenesis of motor neuron diseases, such as amyotrophic lateral sclerosis and spinal muscular atrophy. And in this case, the immune cells can have either a protective role or directly be involved in causing motor neuron death. So the reason why we think that these studies are important is, for example, just in the UK, there's 50,000 reported cases of a spinal cord injury, with 2,500 new cases being reported every year. And spinal cord injury is one of the main causes of paraplegia and tetraplegia, which these conditions have huge costs for the health systems, and there's no treatment currently to treat them. And basically, the devastating consequences of spinal cord injury are due to the fact that mammals have very limited regenerative capacity. So what we hope is that by studying regenerative animal models, we hope to understand a little bit more about the mechanisms of regeneration and devise new regenerative therapies. On the other hand, motor neuron diseases are a group of diseases that are classified as rare. However, when we put them all together, we see that they affect 1 in 300 people. So they are not so rare. And basically, these diseases, they cause the death of motor neurons, which are the neurons that innervate the muscles in the body and control the movement. So what happened in these diseases, motor neurons die, and this causes the waste of the muscle as well. Some of these diseases are very severe. ALS, for example, has a rapid progression, and patients tend to die 3 to 5 years after being diagnosed, and currently there's no treatment for these conditions. So our approach is to understand the mechanisms of regeneration and neurodegeneration is basically to use an animal model, which is zebrafish. And we want to use zebrafish because it has an extraordinary ability to regenerate most tissues, such as the heart, the eyes, the brain, and the spinal cord. Also, this system has also many other advantages that make them an ideal model for our research. For example, they produce large clutches of transparent embryos, which are very easy to manipulate and to image. So amongst the techniques that we use commonly in the lab, we have CRISPR-Cas9 genome editing, which we use routinely for generation of animal models to generate animal models of human diseases like motor neuron diseases, and also to generate genetic tools to study tissue repair. We use quite a lot of live imaging, which is one of the advantages of zebrafish, as I was saying, and these allow us to visualize the repair processes or inflammation in vivo as it happens. And also we aim to use a multi-omics approach, including transcriptomic and proteomic analysis, in order to understand the molecular mechanisms of tissue repair and neurodegeneration. So using one of our regeneration models that basically uses zebrafish larvae to study the spinal cord regeneration, what we have seen is that by causing by completely sectioning the spinal cord in fish, this completely paralyzes the fish, but these zebrafish can regenerate and they recover motility in only 6 days. So we have seen, using this model, that a family of proteins, the F proteins, this family of proteins are involved in cell-to-cell communication. And these proteins are reactivated shortly after injury. And interestingly, one of these proteins, FA4, appears to be highly expressed in leukocytes that are infiltrating the injured neural tissue, so suggesting that FA4 might be important for the process of neuroinflammation. And also interestingly, using CRISPR-Cas9, we generated mutants for all these different F proteins, and we have seen that several mutants display a faster recovery after injury, which suggests that F proteins could be negative regulators of neural regeneration. We also found, using different models of regeneration, that Fs are involved in the regeneration of other tissues. And for example, in this case, we look at the repair of the larval fin fold, which is this caudal structure. And after amputation, the fish normally regenerates in just a few days. But in the mutants, we see that regeneration is not taking place as it should. So this strongly suggests that in this case, FA4 is required for generation. And basically, it seems that F proteins could be key regulators of tissue regeneration. And finally, a recent finding suggests that FA4 could be involved in the pathogenesis of motor neuron diseases. So these diseases are caused by gene mutations, for example, mutations in SOD1 and SMN1 fish. These mutations are linked to amyotrophic lateral sclerosis and spinal muscular atrophy. And recently it was found that FA4 is one of the main modulators of ALS. And for example, patients and animal models that have FA4 mutations survive longer. Interestingly, in fish, we see that when we induce mutations in SOD1 and SMN1, this causes axonopathies that you can see in this panel here. And these can be restored when we also mutate FA4. And the reason why this is interesting is because we know that axonopathies are an early sign of the disease. So before observing motor neuron death, we can observe in patients and in animal models axonopathies. So we think that by using our model, our zebrafish model, we can elucidate the mechanisms of interaction between FA4 and these motor neuron disease genes, and then we can shed light on the yet unclear mechanisms of pathogenesis of these devastating diseases. And that's all from me. Thank you very much.
Thank you very much, Jordi. Well, that's the last presentation. Thank you very much to everybody for this fantastic event. Thank you very much to Sergio and Silvia, who had coordinated and do all the planning and all the models for the program, and also establish all the connections and the live stream in YouTube. And well, thank you everybody for the participation. I think it is just over 7 minutes. So I think it will be okay if we leave it here, so we don't go over time too much. But in the program, we have the emails of everybody, so we can get in touch with each other via email. And one of the things that I'm going to do is to talk with our support office, grant support office in the university, to try to, if they can get some information about grants between the Dominican Republic and UK institutions, because sometimes there are targeted grants to apply between the two countries. I know Royal Society and others, even if it is grants only to establish collaboration. So I think that will be a very good point of starting independent collaborations between groups. And as you know, we are very happy to receive your master students to do some work in our collaboration. We will establish exactly the conditions for that if anyone is interested. And the same when you have a completely functional laboratory, I'm sure there will be students from our university that will be very interested to go there. So Sergio and Silvia, if you want to add anything, please.
No, I just want to thank to everyone for participation, and I hope that we can keep in touch for collaboration working together. So thanks very much. Sylvia is there.
Yes, thank you all for coming today and for being here and for taking the time to reduce the talks to 10 minutes. We know it's really small time to talk, but it was nice to have you all. If you did longer, if you would have taken longer, we couldn't have make it fit into these 3 hours, so we would have needed more time or several days. But we expect it was enough for all of you to know each other, to know what we are working on here and there, and just keep in touch. We will try in a long term to do another talk maybe to see the advances in each project. And thank you all again. Great work, everybody, and we hope you enjoy it.
Okay.
Thank you, everybody, and keep in touch, please. Thank you very much. Bye-bye.
Bye. Bye-bye.
Thank you, everyone. Goodbye.